மைக் இந்த மைக் தான் ரெக்கார்டிங். ஹலோ சார், இது கேக்குதா சார்? ஓகே சார். அது ஓபன் ஆகும் போது எல்லாம் பேசவே இல்லை. அருண், ஏன் உனக்கு தராத வேலை? இப்படியே வச்சுட்டு ஒரு கையில மைக் வைக்கணும், ஒரு கையில இது வைக்கணும். போர்டு சேஞ்ச் பண்ணிட்டு வேற ஏதாவது போர்டு எடுக்கணும். அருணே அச்சா போர்டுல எடுத்துக்கோடா. ஜூலியர் ஏதாவது ஜூலியர் போயிட்டு ஸ்டாஃப் ரூம் வந்து ஒரு போர்டு எடுப்போம். ஒரு போர்டு
கிராத் செகண்ட் இயர் பேரண்ட்ஸ் அவங்க எல்லாம் பண்ணாதான் அடுத்த வருஷம் இருப்பாங்க.
Sir, Samitai,
we welcome Secretary Sir Praveen ஒரு ரெண்டு மூணு பேர் சேர்த்து secretary and correspondent and the chief justice will raise the அத கேட்கதான் போயிருக்காரா செகண்ட் ஆனா ஓதுறாங்க சார். ஜூம்ல தானே சார் வராரு? ஜூம்லயா? ஓகே, அதுக்கு தான் சார் ரெடியா இருக்கு பாருங்க. இல்ல அவங்க லிங்க் எல்லாம் வந்துருச்சு சார். அவங்களுக்கு அங்க இருக்கு, அங்க அங்க இங்க சார், இங்க நேஷனல் சயின்ஸ் டே வாங்க. அதுவா? அது இல்ல சார், இருங்க இருங்க சார், யூடியூப் தானே சார்? ஆமா சார், ரெடியா இருக்கு சார். சார் நான் யூடியூப் தான் சார், லிங்க் யூடியூப் தான் சார். சார் அது லிங்க வச்சு டவுன்லோட் பண்ண லேட் ஆகும் சார். யூடியூப் டவுன்லோட் ஆகும் சார். சார் யூடியூப் வந்து டவுன்லோட் பண்ணா அதுக்கு 2 ஆப். அந்த ஆப் டவுன்லோட் பண்ணி எத்தனை சார் மெயில் லிங்க் தான் சார். டைரக்டா யூடியூப் ஆனா உமார் சார் கிட்ட இருக்கும் சார். ஆமா சார், நீங்க அனுப்பின வீடியோ சார். இல்ல சார், யூடியூப்ல டைரக்டா
1 2 3 4 5 6 7 8 வீடியோ இருக்கா?
மூணாவது என்ன ஏதோ ஓடுது, இதுதானே? இது வேறயா? லிங்க வந்து மெயிலுக்கு காப்பி பண்ணிடணும். அதுக்கப்புறம் இல்ல வாட்ஸ்அப்ல டைரக்டா இருக்கு சார். அதுல அப்படி போகணும் சார். லிங்க் தான் உன் பேர் தான் இருக்கு. என்ன ஒரு பிரச்சனைனா விவகாரம் பிரச்சனை மெசேஜ் அனுப்பிட்டே இருப்பான்.
பூபதியும் மோனிஷும் பாடிப்பாங்க. சிடி ரிலீஸ் சார். இது சாவனேர் தான் சார். ஸ்டார்ட் தான் சிடி அப்ஸ்டார்ட் தான். ரிலீஸ் ஆஃப் சாவனேர் தான் சார். அந்த பையன் அதான் ஃபர்ஸ்ட் இயர் பையன் சார். செகண்ட் இயர் பையன் அந்த செகண்ட் இயர் பையன் வர சொல்லுங்க.
சார் இது டாக்ஸானமிக் எல்லாம் போட்டு டாக்ஸானமிக் எல்லாம் ரெடி. ஆமா சார், இப்போ ஒரு மெசேஜ் அனுப்ப சார். உமர் சரி சார். சப்டைட்டில் சொல்றேன். எது சார்? செகண்ட் கேஸ் செகண்ட் இயர் அல்ரஷீதா இவரே ஃபர்ஸ்ட் இயர்
என்ன கிளாஸா என்ன ஊத்துறா அவர
அதுக்கு இங்கிலீஷ் மீடியம் சொல்றேன். ஆனா இந்த போட்டோ
இங்கிலீஷ் மீடியம் வேணும்னா நான் ரெடியா இருக்கேன். என்னன்னா ஆப் தரேன். இங்கிலீஷ் மீடியம் பார்த்து சொல்றது. இங்கிலீஷ் மீடியம் பார்த்து
ஆமா போயிட்டு விட்டுருங்க.
முன்னாடியே அவங்கெல்லாம் மாப்பிள்ளை
போட்டோஸ் எல்லாம் போட்டு விடுவோம்.
இதெல்லாம் இதுலயே சேவ் ஆயிருக்கு. இதெல்லாம் ஏன்னா யூஸ் கல்வி விட்டா கேஸ்
தமிழ் தாய் மதர் நீர் அது நான் குழப்பம் ஓகே தமிழ்ல சொல்லுவாங்க
நல்லா இருக்கு. இது 40 நிமிஷம் அது. இதுக்குதான் ஃபர்ஸ்டே வந்து இதெல்லாம் டவுன்லோட் பண்ணிட்டு என்ன ஆடு வந்து ஏன் கண்ட்ரோல் இல்ல, ஆடு அவங்க கண்ட்ரோல். எதை ஓப்பன் இல்லையா வச்சுக்க.
ஃபைல் நோட் பேட் எல்லாம் அங்க இருக்கு இல்லையா, நம்ம ஃபைல் நோட் பேட் எல்லாம் அங்கே இருக்கா இல்லையா, கிடைச்சிருச்சா வாங்கணும்.
அங்கே நீ வேற என் போட்டோ என் வாட்ஸ்அப்க்கு எல்லாத்தையும் அனுப்பி வச்சிருக்க.
இப்ப அது போட்டு பாப்பீங்க நான் என்னத்த பண்ணேன்
இல்ல விலைக்கு போட்ட என் வாட்ஸ்அப்ல வச்ச கேஸ் ஆகும்.
நம்பரே தெரியும்
ஒரு செகண்ட் வீடியோ
என்னது
நம்பரே தெரியாது, என் பேர் வந்து என் பேர் வராது.
சேவ் பண்ணதுக்கு அப்புறம் எப்படி பண்ண முடியும்?
நம்பர் சேவ் பண்ணா
நம்பர்லாம் பெரிய பிரச்சனை வராது. எனக்கு ஹாஸ்டல்ல பிரச்சனை வரும். சரி இது எப்படி ஃபுல் ஸ்கிரீன் வராது, வாட்ஸ்அப்ல இருக்கிறபடி இது வராது, ஃபுல்லா வராது.
அப்படியே ஒரு போட்டோ எடுத்துட்டு இப்படி அடிச்சு
அது வராது.
சரி வீடியோ
போட்டோ வச்சு
ஃபுல்லா கொடுக்கணும்னா ஃபுல்லா வரும். இல்ல அது ஆப் போய் கொடுக்கணும்.
வீடியோ பண்ணல அது போட்டு
ஒரு விஷயத்துல ஒரு டைம் தான் சொன்னா பரவாயில்ல. ஒவ்வொரு விஷயத்தையும் ஒவ்வொரு மாதிரி வச்சு நீ செஞ்சு நான் எதை போடுவேன். இல்ல இதெல்லாம் ஆல்ரெடி சேவ் பண்ணி வச்சிருந்தா பரவாயில்ல. சரி அசலை கூப்பிட்டு அது வந்து எந்த எல்லாத்தையும் ஒரு கூட்டத்தை அமைச்சிருக்கோம். எங்க இருக்காங்க வீடு? தம்பி அஜீஸ் இருக்கானா? இல்ல அங்க அனிமேலியா இதை பயில எந்த பொருள் வச்சிருக்கீங்க? இதை எவனா இருந்தானா பாத்து கேட்டு
பிஸ்கெட்டா பிஸ்கெட்டா பிஸ்கெட் சிக்கனா உஸ்கே சிக்கனா
சிக்கன் பத்துமா
இருக்குமா? தமிழ் தாய்
உணர்ச்சி சாப்பிடாது. போன மாதிரி டெலிட் பண்ணிட்டா எனக்கும் கண்டிப்பா தான் இருக்கு.
வெளிக்குள்ள நிறைய வந்தா எங்க இருக்கு
வெளியே போயிட்டு அதில் கழிப்பு டைம் வந்து
நீங்களாம் ஏதாவது சொன்னீங்கன்னா அந்த மாதிரி வீடியோ வந்து போடுவேன். இதான இது போலவா இப்ப வேர்ட்ஸ் வர அமைப்புமே சொல்லி வரணும். பிளான் பண்ணி சொல்லணும். தம்பி
esteemed laboratories, universities and research institutes. നമുക്ക് ടെൻഷൻ ഇല്ല.
அதான் மிஸ்டேக் சொல்றீங்களா? அப்படியே பாதியா பர்சன்ட் நான் உங்களுக்கு பண்ணேன். ஓகேவா? அந்த மாதிரி
நானொரு ஊமை சார், எனக்கு பேச முடியாது. ஊமை எதுக்குப்பா வந்து
this is not a communication.
as per the present context. There was number of senior science, number of senior science, number of senior science.
சார் இது நான் அதிகமா பேசுற மாதிரி வருது சார். பேசாத மாதிரி இந்த இமேஜ் வர வீடியோ அதுல பேசுற மாதிரி இல்ல, இந்த மாதிரி வேணுமா இல்ல பேசுற மாதிரி
சீக்கிரமா
இது 4 நிமிஷம் இது முடிச்சதுக்கு அப்புறம்
என்ன பேசு
ஏற்கனவே பெரிய வேலை சார். நைட் டீ பார்ட்டி இருக்கா சார்?
இது பிபிசி சார். இப்ப
சார் சார் நைட் டீ பார்ட்டி இருக்கா? போஸ்ட் டீ பார்ட்டி, போஸ்ட் ஃபங்க்ஷன் இதுக்கப்புறம் சார் நைட்
லாஸ்ட் நமக்கு பார்ட்டி
வேணுமா?
வேணும்.
படிச்சு படிச்சு படிச்சு
வராங்களா?
இது பண்ண வேண்டியது நான்
எல்லாரும் அவங்க வேலையை பார்க்க போங்க. எல்லாரும் அவங்க வேலையை பார்க்க போங்க. படம் முடிஞ்சு இவற்றெல்லாம் இந்த வீடியோ போனவங்க எல்லாம் வீடியோ தான் கொடுக்குறீங்க. அரை மணி நேரம் பேசுறேன்.
படி எல்லாம் கரெக்டா தான்
வெல்கம் அட்ரஸ் எங்க உடனே தானே இருக்கு?
இல்ல ஒரு பேப்பர் மட்டும்தான்
வெல்கம் அட்ரஸ் எங்க? Presidentially, honoring it, inaugurally. Sir, felicitation கெல்லாம் முடிஞ்சதுக்கு அப்புறம் தான் எல்லாரும்
felicitation முடிஞ்சு honor பண்ணிட்டு சோகம் பண்ணிட்டு அதுக்கப்புறம் தான்
presentation பண்ணிட்டு இந்த felicitation
பண்ணிட்டு அதுக்கப்புறம் திரும்ப அந்த பேஜ்
வந்துட்டாங்க.
ஏய் செக்ரட்டரி இருக்குடா
புதுசா இருக்கு. புதுசா வந்திருக்கிறா.
வேற வருஷா யார்ல அது
ரொம்ப பெரிய சயின்டிஸ்ட்.
சரி
வீடியோ
Assalamu alaikum. Honorable Dignitary, beloved head of the department, respected professors and students, very good morning to all. It's my pleasure to extend a cheerful welcome to you all to the third international conference on environment and health, conducted by Department of Zoology, Jamal Mohamed College. Tiruchirappalli, the heart of Tamil Nadu, where the golden sun shines, a land rich in sacred culture. On one side, the largest stupa in the world, and on the other side is blessed by river Kaveri. Our Tiruchirappalli has witnessed many historical moments. The Tiruchirappalli houses its own historical monuments like Srirangam, the world's largest working temple, and the Kallanai Dam, the oldest dam in Asia, which is made up of stone, makes Tiruchirappalli absolutely amazing. One of the wonders of Tiruchirappalli is Rockfort. It is the most loved place of the people. We welcome all the participants from various states to the Rockfort city of Tamil Nadu.
Jamal Mohamed College started its journey on 1951, an autonomous institution affiliated with the Garidas University, Tiruchirappalli. The college is formed by the two great philanthropists, Haji and Jamal Mohamed Saib and Janab N.M. Kashamin Rao. It was established as a religious minority institution with the primary objective of providing higher education to the downtrodden and socially backward section of the society in general and Muslim minority in particular.
The Department of Zoology is one of the pioneering research departments. The UG and PG programs affiliations during 1957 and 1969 respectively. The department was elected as the first research department in 1974 by the University of Madras. Since the last 2 decades, the department has also been offering career-oriented programs in medical lab technology approved and recognized by the UGC New Delhi. Since 2020 to 21, the UGC also granted permission to offer vocational courses under NSQF scheme. A 1-year diploma in aquaculture, and we also offer short-term value-added courses such as ornamental fish culture, bird keeping, dairy farming, and vaccine technology. The department is supported by DST Physics program and chosen as one among the 5 science department in college for DBT Star College scheme. A remarkable number of research projects have been conducted by our faculty with financial assistance from agencies such as UGC, GAE, INSA, GST, PARC, and MOEM. The department so far has produced 52 PhDs and our own department is only 5 patents on interdisciplinary areas.
As per the present conference is to focus on the latest research on environment and health. This will serve as a good platform to gather a number of senior scientists and experts from esteemed laboratories, universities and research institutes in India and abroad. Young scientists and students can utilize this as an opportunity to have effective interaction with eminent personalities and to understand the latest research advancements in the frontier areas of animal science and environment. So once again, we welcome all the dignitaries, professors and students from various colleges to the International Conference on Environment and Health.
Before we get into the program, it's always good to start with a prayer. It is true that a prayer that comes from the heart can move mountains. It increases your faith and brings you closer to the God. I would like to call Mr. Mohamed from ICEH Zoology to recite few verses from the Holy Quran.
أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. إذا زلزلت الأرض زلزالها وأخرجت الأرض مثقالها وقال الإنسان ما لها يومئذ تحدث أخبارها بأن ربك أوحى لها يومئذ يصدر الناس أشتاتا ليروا أعمالهم فمن يعمل مثقال ذرة خيرا يره ومن يعمل مثقال ذرة شرا يره. السلام عليكم ورحمة الله وبركاته.
Thank you, Mr. Hayo. I kindly request everyone to raise for Tamil Thai வாழ்த்து. தமிழ் தாய் வாழ்த்தினை பாட மோனிஷ் மற்றும் முதுலிங்கர் பூபதி ஆகியோரை அன்போடு அழைக்கின்றோம்.
சீராரும் பரதம் எத்தனமும் அதிசிறந்த அத்திரகர வாசனை போல எத்திசையும் புகழ்மலை தமிழனுக்கே உன் சீர்தமன்
Thank you. If your actions create a legacy that inspires others to dream more, learn more, know more, and become more, you are a leader. I warmly welcome our respected head of the department, Dr. I. Joseph Anthony Gerald, sir, to welcome the gathering.
All praise and glory be to the Creator. In the name of Almighty, the Most High, I begin. Wishing you all a blessed morning, ladies and gentlemen. Most esteemed Secretary and Correspondent sir, Member and Honorary Director, Dr. Nihar, or respected and beloved principal, versatile chief guest of this international conference, Sri J. Justin Mohan IFS, National Biodiversity Authority, and to be shortly the Principal Chief Conservator Forest Government of India. Is also a relatively talented, so we can give a warm clap, please. The Bachelor in Physics in our sacred quarters. Dynamic Directors of Hostels, Dean of Arts, Science and Research of Barsat, the organizing secretary of this two-day program, Professor SS, our guests of honor, Dr. Atkusha Sara Mahaguru, our former professor, Dr. P. Shalvani sir, fellow professors, beloved former teachers, learned colleagues, faculty members from sister departments and neighboring colleges, research scholars, non-teaching staff, invitees, guests and press persons, participants of this conference, my dear student friends in Jamalians, ladies and gentlemen, Assalamu alaikum and a very good morning to all here. I am honored indeed to extend a warm welcome to each one of you assembled here and listening to us online through the e-gadgets. I am delighted and truly honored to welcome you all to this third International Conference on Environment and Health. I think this scientific venture will be a hallmark achievement as this conference is one of our earnest attempts that will help combat the bottlenecks and expand the knowledge among researchers, scientists, and life science professionals, and enable them to the next level of understanding and information on environment and health issues. Yes, friends, there is a growing concern about the links between the environment and health. Worldwide, 1/4 to 1/3 of the burden of diseases appears to be because of environmental factors. Of course, the vulnerability and exposure among individuals vary. There is reasonable understanding of the cause and effects and the relationship between pollution in water, air, and soil impacting human health. Health consequences are also due to environmental factors resulting from climate change, global warming, and the other environmental factors and chemicals that too disrupt endocrines on humans as well as wildlife. Scrupulous disposal of waste, overexploitation of natural resources, and every activity of this kind leads to health implications such as respiratory, neuroendocrine, neurotoxic effects influencing the onset of puberty, lifestyle diseases, and obesity and diabetes, even among children. Friends, I am sure this conference will focus on all these issues, and I congratulate our organizing secretary, Professor Shahid, for having identified this hot topic of present concern and arranging for this scientific conference of eminent scholars and researchers at our holy soil in Jamal. I give it a great pleasure to intimate at this juncture that almost all speakers on this conference of today and tomorrow are alumni. Thanks to the vision from our founders of this institution. I take this opportunity to welcome to our midst the most honorable secretary and correspondent sir, who always has a keen interest on our department and its welfare. We welcome you, sir. We are proud to welcome our distinguished research advisor, our Mahaguru, as I said, Dr. M.A. Akbarshan, sir, who also served in our college and thereafter went to the university to extend his services. And he is again back to our college. He is assisting us as a research advisor in the Atlanta vaccine project. So we welcome you on behalf of the department of the college. We are truly blessed to have our former professor, Dr. P. Shawnami, who is the main reason for inculcating research in our college campus. It is he who brought in Jamal College in the map of nuclear science and atomic research. We welcome you, sir, on behalf of the department of the college. Any activity in the campus and the success of Jamal is due to the inspiration and the positive vibration we invite from our learned principal, who commands a great deal of respect and admiration from all the faculty, staff, and students of our college. We welcome our principal. Well, friends, I'm truly happy and proud. Want to introduce and welcome our guest speaker, the man of the day, Shri J. Justin Mohamed. He belongs to the 99 batch of IFS service staff who was trained in the Indian Forest Service at the Indira Gandhi National Forest Academy, Dehradun, and thereafter the Indian Military Academy, Dehradun, and the Lal Bahadur Shastri National Academy Administration, Mussoorie. He joined as secretary to the most prestigious scientific body, National Biodiversity Authority (NPA), in September 2019. He was heading the Kerala Soil Survey and Soil Conservation Department, Groundwater Department also, in various capacities. And sir has served as the highest authority in the various forest departments of Kerala, and has also worked in Munnar, Palkar, Silent Valley National Park, and in various other places. He has served as a project director of rupees 1,840 crores of rupees in Kerala, one of the projects in the agrarian ecosystem that spread across 3 districts of Kerala. So the most important of all the services to the people, I think, is this. By the government of India in the year 2011, Sir was appointed as the first secretary to the High Commission of India to go to Colombo to coordinate the relief and rehabilitation and development of war-affected families in Sri Lanka. During his 3-year tenure there, he had implemented 50 major projects, worth rupees 7,000 crores in Sri Lanka, including the Indian housing project involving construction of more than 50,000 houses for the benefits of the conflict-affected communities, such as the human scientists of today. As an ordinary little boy, he started his schooling at Campion, who lived in our own city, grew up to this collective achievement due to his sincere hard work. Sir has graduated in agricultural microbiology well before he was in our campus, wanting to graduate in physics for 2 years and thereafter moved over. And Sir has also earned a bachelor degree in law from the National Law School of India, University, Bangalore. He holds a master's degree in management and public policy from Indian School of Business, Mohamed, and an MBA degree from Alagappa University as well. Recipient of various awards, and such is our eminent scholar who is with us this morning to inaugurate our international program, and on behalf of the college management and on our personal behalf, we extend you, sir, a very warm welcome. First thing, before I conclude, I beseech you all, the participants, to utilize this opportunity and pave way for good academic networking, to aspire for high quality research and be motivated with a wider set of strategies to pursue. I wish you all very purposeful days ahead and eventful stay at Jamal. Make the best use of the conference, friends. Thank you. Now, before I finish, I wish to add to our guest speaker, Dr. Sri J. Justin Mohamed, that twice he was chosen for IPS cadre as well. But then he wanted to take up science as his venture and got into his IFS service. So we are really impressed and inspired the way sir has graduated and has achieved his credentials. We are very happy and we extend a warm welcome to you, sir. We welcome our former president of the ICEH-2023 who has graced this morning. Welcome you. Thank you, ladies.
Thank you, sir, for your nice and warm welcoming. The great leader are almost always great simplifier, who can cut through arguments, debate, and doubt to offer a solution that makes everybody can understand. Let us now welcome the man who doesn't need any introduction and known among all of us for his wisdom and knowledge. I welcome our honorable principal sir to deliver the presidential address.
Bismillahirrahmanirrahim. Most honorable Secretary and Correspondent, respected Federal Department of Zoology, Dr. Joseph Jamal sir, respected former professor and present honorary research advisor, Dr. Akbar sir, respected Dr. P.S. sir, former Federal Department, former professor of Zoology, Dr. K.S. sir, and most honorable today's chief guest, Shri J. Justin Mohan, IFS, former secretary, National Biodiversity Authority, Government of India, respected Dean of Science, Dean of Research, faculty members from the Department of Zoology, from the Department of Botany, and other faculty members, participants who are participating physically and through online, and faculty members from other colleges, my dear young Jamalians. I am very happy and appreciate that the Department of PG and Research Department of Zoology is organizing 3rd International Conference on Environment and Health. Even though they are mentioning that 3rd International Conference on Environment and Health, the Department of Zoology, they are undertaking research on environment and health, as far as I know, I think from 1980s. So from that, those days onwards, they are doing, the Department of Zoology is doing research on environment. I used to hear the word a lot, many times from Dr. P. Shaulavi that the environment, the radiology, Kaveri River Project, atomic energy, Baba Atomic Research Center, Dr. Edward from Bishop River College. I used to hear these words from Dr. P. Shawarame for many days. So zoology department is known for research. I used to tell this. Even though in the alphabet, Z is the last alphabet, the Department of Zoology is number one in Jamal College. A lot of research from those days onwards, they are doing that. The legacy is continued now, even now. And because of the Department of Biotechnology, it recognized our college. 5 of the department are recognized by the Department of Biotechnology continuously for the 3rd time. Today, I think after PSCKR college, our college is honored by DTT. Our college is the only college other than PSCKR. Ours is the only college. We are honored by continuously by 3rd time. That is, we are recognized by the Department of Biotechnology. Because it is very difficult to follow the rules and regulations laid out by the Department of Biotechnology. But our department, the Department of Zoology, Mathematics, Physics, Chemistry, and Botany, they are bringing knowledge to the total college. So in that way, I congratulate the Department of Zoology, and I am very glad that because of these projects, the basic science, the basic research have been inculcated in the young minds, not only the college students, even to the adopted villages. The school students also are getting knowledge, getting research ideas from the fundings of the Department of Biotechnology. So now the Department of Zoology is organizing the international conference, 2 days international conference on environment and health. So environment and health both cannot be treated. If environment is good, and always health will be good. So the topic chosen by the Department of Zoology is also very nice, and definitely the outcome of this research will reach the global level, and the interactions. The international conference, we don't expect thousands of students participation or thousands participation, and many papers presented. Even it is few papers, 1 or 2, but the paper should be on par with the other excellent papers published in Nature and other journals. That is what we expect. That is why the money is spent. Even the UGC and Government of India, they are reducing the amount for research. But I am very happy that I appreciate our management. I thank our management on behalf of all the staff members. The management is providing money for organizing such international conference. Management is providing 50,000 rupees for the department to organize such conferences. And also I am very glad that the management is providing seed money to the professors to do research. Not only to the professors, the management is extending hands to the students also for their ideas, for innovation. So I request all the participants and the student friends, think, analyze the problems faced by the common people and try to provide solution to all the problems. That is what expected from any national or international conferences. That is what we also expect. Wish you all the best. Thank you very much. Thank you for your priceless words, sir. A good leader leads the people from above them. A great leader leads the people from within them. With my open arms, I welcome our esteemed secretary and correspondent, Dr. A.K. Khaja Naseemuddin sir to the stage for a few words.
Bismillahirrahmanirrahim. All praise be to the Almighty, the most beneficent and most merciful. Today, the PG and Research Department of Zoology of Jamal College are organizing third international conference on environmental health. In the most auspicious presence of our chief guest of today, Sri J. Justin Mohan, IFS former Secretary, National Biodiversity Authority of the Government. And our professors on the dais, Dr. Abarsha and Dr. Saurav, and the head of the Department of Zoology, and our dynamic principal, and all the members of the staff are present over here, and the participants and students. It's a 2-day workshop. That is a welcoming one. I have to congratulate the organizing secretary. It's Sheikh Umar Sari, Associate Professor of Zoology at Mohamed College, and organizing secretary today in this program. Pollution means something we are aware only in the later stages of all things happen. At the early stage, it was not to the knowledge, it was not aware of it, and everything was the innovations of technological applications, everything came over. But later on stage, in each and every event, there was a pollution, and it has to be eradicated. And that is why research has been followed, and it has been widespread throughout the country amongst the scientists, as well as all the newly budding scientists who come over from the education department and the various parts of the studies, such as physics, chemistry, and zoology, and all the other conventional science. Anyhow, I'd like to say that a small thing here over here, there is information regarding water pollution, air pollution. The water pollution is throughout the country. It cannot be totally eradicated. And it has been observed, and many steps have been taken to prevent the pollution in the water, even in the groundwater, and all the industries which have been emitting pollutions to the river beds and all kind of lakes and water tanks and everywhere. At the same time, the air pollution is widely distributed among the entire country, entire global. And all of you know that the global warming is there. That is also one of the solutions. And what will be happening, the consequences when the global warming has been taken over? And many things are being changed. Even the sea level, which has been raised, and it is higher than what it was to be. So how far these scientists have been actually, we can just imagine on it. We have to thank the scientists who have been taking so much of pains to discover what is being happening in and around the environmental factors and what are the pollutants and how to be eradicated in all the events which have been taken throughout the country. And the government has been taking a keen observation, and the pollution control group of central and state has been established, and it has been followed thoroughly. And all of you know that nowadays there's even the automotive mobile, they have been taking the steps to what we call control the efficient systems of the pollutants. So nowadays you cannot simply make any manufacturer a vehicle that can come out easily in the market. Likewise, everything is there. Imagine the air traffic. You know how much of white petrol fuel has been consumed, and the gases which has been spread in the air. And we see the network of the entire, what we call as the air traffic network throughout the globe. There is temperature, raising temperature in the air, and also the pollution is being widespread throughout the globe. And how it is being affected, and what are things that have the impact over it, it is being measured, and slowly it has been also coming to a control. What are the fuels which has to be added in the aviation, as well as the automotives and everything. Even the ships, which the earlier stage, there was a very polluted types of engines, which was even polluting the sea level area. So everywhere, what all things are happening as a mechanical foundation, it has been established to be observed and see whether how much are polluted and done. Anyhow, the total eradication of pollution cannot be controlled. The percentage may be minimized, and the entire 100% of what we call as pollutant eradication will not be done. Somehow it will be a little bit of what we call as a reduction of percentage. So likewise, this education is here, and I find that many speakers from the technical persons have come over here, and I think they will give a wide picture about the what we call the future generations, emissions and control. And myself, as a commerce student, I also had the intensity to make the participation as a science candidate. And in my industry, I myself made a zero discharge system of the other industry, along with an engineer, consulting engineer. And I succeeded, and the only industry which I was having in southern India had a zero discharge. So that was a pilot plan, and it was very successful. So like that, everything is innovative, and it depends upon the person who will take the interest to be make the study over it. So even our professor, Mir, sir, Anis Mohamed has come over to our industry, and he did the PhD in the environmental pollution control. And many times, Dr. Sir has made the meetings and conferences and all the get together to make the conversion of all types of water level and even the water pollutants and everything, the study which has been taken by him. And now also he is very active on that part, even though he left the college earlier after getting his retirement, and he is continuing to do the same for Dr. Agosha also. So we have to congratulate them on the graduation. And our friend, Justin Mohamed, is 10 years junior to me in the school. 20 years actually. So I am very glad that he is the alumni of our school as well as our college also. So both of us are having the relativity of alumni, and therefore I consider it in the occasion. And very happy to see him also studying in the same school and studying in the same college. Well, sir, thank you. So with these few words, let this program go on to later heights and make the achievements of pollution controls and systems, and the student friends have to take care of it. It is here, it is everything on your hand, where you have to take the reality of life there. So with these few words, I conclude my speech, invoking the blessings of the Almighty to shower the chosen blessings on all of your endeavors. Thank you.
Thank you for the great speech, sir. Next is honoring the guest. I welcome our esteemed secretary and correspondent to honor today's chief guest, J. Justin Mon IFS, with the adorning of show.
Now, it is the great time for releasing the souvenir. We request our secretary and correspondent to release the souvenir and the chief guest to receive it. We request all the dignitaries on the dais to join, please.
Thank you, sir. Every moment is a fresh, new beginning, a wonderful inauguration of the great cosmic journey through the universe. We can do whatever we want. We can change the reality at any moment. On behalf of the Department of Zoology, I welcome our chief guest, who is a former Secretary of National Biodiversity Authority, Government of India, Mr. J. Justin Man, IFS, to the stage for the inaugural address.
Thank you very much. Most respected Secretary and Correspondent Dr. Raja, sir. Principal of our esteemed college, Dr. S. Ismail Muhyiddin, sir. Head of the Department of Zoology, Jamal Mohamed College, Dr. Joseph Jamal, sir. Dr. P. Sahul Nabil, sir, who has been a great inspiration even for me. Dr. Akbar Shah, sir. Organizing Secretary, Dr. Sheikh Kumar Shahid, sir. Professor Anis Mohamed, Professor Sadiq Bukhari, Mr. Saeed, esteemed professors of the Department of Zoology and other departments who have assembled here. My dear students, the Department of Zoology and other sister departments who have assembled here. Invitees who have come from other colleges. I am extremely happy to be in Jamal to attend this program almost after 10 years. I came to Jamal last for attending a program on 25th June 2012 at the invitation of the college to inaugurate the civil service exam study center. So that was the time coming here after 10 years. So when I came last time, I was very curious to go and sit in my class. I studied physics on the other side of the campus, but I was very surprised to see the development that have happened over the years. And this time when I saw the same area, I was actually so much thrilled to see the infrastructure development that has happened in Jamal for the last 30 years.
Thanks to a very, very vibrant and progressive and forward-looking management of Jamal, and also the non-alumni who have been supporting Jamal financially as well. Today, Jamal students are found across the world. In fact, when I was heading the agriculture department in Kerala, I was asked to report to a person who was assisting the minister. And when I interacted with him, he was telling that I am also from Jamal. He was serving as special secretary in the secretary of the government of Kerala. So you can see Jamalian across the world, whether it is in Europe or Middle East, or Canada or the United States. So thanks to their contribution as well, the college witnessed enormous development over the years, and apart from the infrastructure development, which I'm seeing the last 30 years, we were studying an asbestos. The roof of our class was made of asbestos. It was very low lying those days, but now fantastic building, lot of very good infrastructure, AC hall, terrific development. And, apart from such infrastructure development, we are also looking for research opportunities. I am so happy that the Department of Zoology alone has produced 56 PhDs in the recent past. And lot of collaboration is happening with different scientific bodies, whether it is the Department of Biotechnology, Department of Science and Technology, PAS, Baba Atomic Research Center, various international collaboration. So I am seeing so much of it's a happening college. Jamal has always been very lively. He always brings life. Those days we used to tell it is Jolly Jamal because there is a lot of life. There is always saying that you go to Joseph, it is like jail Joseph. You come here, it's a Jolly Jamal. So, so there is always life in Jamal. Students are happy. Teachers also make you happy. And people who are going out of Jamal, fortunately, they all settle well in their life. So I am here today to address you after this inauguration program. I am going to give you 2 lectures. Very elaborate preparation I have made. One technical subject on how biodiversity is linked with environment and health. So I am sure that it is going to be really informative for you. But the post lunch session is going to really help you a lot, because nowhere you are going to see that type of information in any YouTube or anywhere, because it is out of my own personal experience. I'm going to share on career opportunities for graduates. So I want every Jamalian who is passing out from our esteemed college to come up in life, and for those who are having their own family business, trying to most likely take over that business of self-employment, but who are going to depend on a salaried work, those who want to get a job, the government or a private sector. I'm sure that the evening class is going to benefit you enormously. So I'm not going to discuss in detail about environment and health during this inaugural address, because that is going to be taken care after this inaugural session by myself. I am going to start the presentation, 1 hour presentation on how biodiversity is going to impact environment and health. So this conference also, just like how Jamal is going to take care of the research sector, such conferences, particularly international conferences, where we are going to pick a lot of scientists from other countries. Today I am seeing speakers from US and other countries. So these are going to bring in a lot of new ideas. It's going to ignite the minds of the younger generation, the next generation, on the recent advances in the health and environment sector. I'm sure that this 3rd international conference on environment and health that is being organized by the Department of Zoology, Jamal Mohamed College, is going to throw in a lot of new ideas and opportunities for the students as well as the researchers of our great college. I wish you all the very best. Formally, I inaugurate this session. international conference on environmental health. And I wish you all the very best in your life. Thank you.
Thank you, sir, for sharing your expertise. The future belongs to those who believe in the beauty of their trees. I would like to invite Mr. ICEH-2023 sir to propose the vote of thanks.
Good morning, Manila. This is my time to present vote of thanks for this. The president of this conference, we express our sincere gratitude to our secretary and correspondent, our colleague. Thank you, sir. And the director of our college, here, and the assistant secretary of this college, here. Thank you, sir. Then the member and honorary director of Dr. Nigam and the Mohamed Vice Principal and the Mohamed Principal in Asia and the Director of Hospital Administration. Mohamed Fadil in Asia, Hyderabad, Director of Hospital Administration in Asia. I present my sincere thanks to all. I also present my sincere thanks to Dean of Arts, Dean of Science, and Dean of the Institute of Department, Secretary and faculty members, non-teaching staff, and research scholars came from different institutions, and ourselves, students and friends, and the all arrangements made by practitioners and the video conferencing everything, and the guests and the participants, and all I thank you. Thank you.
Now it's time for refreshment. The refreshment will provide outside of the hall. And request you to assemble after the tea break at 11:30 for having today's first technical session. Thank you.
கொஞ்சம் பொறுமை ஓகே
எல்லாத்தையும் புரியும் தமிழ்ல வந்து கேப் நல்லா சைலண்டா நல்லா மெதுவா படி
அந்த கவர்ல போட்டு இல்ல சார் இருக்கு என்ன முடியும் சவுண்ட் போயிடுதுன்னு சொல்லி போடுறது
வேற என்னண்ணா வேற எதுவும் என்னண்ணா
டீ பிரேக் வேணும் அப்படிதான்
நீ என்ன இங்க வந்திருக்க?
ஒரு சேரோடு போடு வைரஸ்
இன்னும் மேலே
ஜாலியா காலாடி
இவன்ட்ட ஏன் மெசேஜ் அனுப்புறேன்? யூடியூப்ல இருந்து டவுன்லோட் பண்ணா ரொம்ப கஷ்டம். விட்நெட் ஆப் வேணும்டா. அந்த பச்சை கலர்ல இருக்கும்.
யூடியூப் டவுன்லோட் பண்ணவா? ஆன்லைன்லயே டவுன்லோட் பண்ணா விட்நெட்.
அது ஆப்ல செய்வாங்க. டெஸ்க்டாப்ல செய்வாங்க. எப்படி ஆகும்? சரி நீ ஆப் வந்து என்ன பண்ற? சாப்பிட போற கிளம்பிடுவேன். சாப்பிட போறது அதெல்லாம் முடியாது.
நான் ஐடியா சொல்லி தரேன்.
ஓகே சரி வழிய விடுறா. சரி நீ ஆப்ரேட் பண்ணிட்டு உட்காந்து நான் பின்னாடி உட்காந்துக்கிறேன். ஆனா பண்ணி பேர சொன்னாரு. இல்லன்னா கண்டிப்பா சொல்றாரு. நீ கேட்ட உடனே அந்த பண்ணி போயிருக்கேன்.
இன்னொரு சேர் தான் போட்டு உட்கார நானும் உட்கார போர் அடிக்கும். இங்க உட்காந்தா நல்லா இருக்கும். அவங்க உட்காந்தா போர் அடிக்கும். அடுத்த செகண்டே மறுபடியும் சொல்லிட்டே அவரோட
நம்ம பேரை சொல்லி நான் பின்னாடி போவா சார் அஜீஸ் உட்காந்துக்கிறாங்க சார் அப்படின்னு. ஏன்டா இப்படி சார் சார்
நடந்துட்டு பேச நல்லா இருக்கும். சரி 5 minutes ஸ்டூடெண்ட்ஸ் எல்லாமே வெளியில டீ பிரேக் சொல்லிட்டு வெளியே போயிடாத. வெயிட் பண்றது செகண்ட் பிஎஸ்சி ஃபர்ஸ்ட் பிஎஸ்சி
சார் கேட்டாரு எல்லாரும் இருந்தாலும் அட்டென்ஸ் போடணும்ன்றாரு. கேரளா பசங்க
நீ போட்டது எல்லாரும் பாத்துட்டியா?
பார்த்துட்டாங்க.
பாத்துட்டியா? இன்ஃபோ பாத்துட்டியா? எல்லாரும் பாத்துட்டியா? இன்ஃபோ பாத்துட்டியா?
எல்லாரும் வந்து அட்டென்ஸ் போட மாட்டாங்க சார். குரூப்ல போட்டு
ஃப்ரண்ட்ல உட்காரா இங்க உட்காரா. சார் ஏன்னா இது ஆப்ரேட் பண்ணுங்க.
நான் மட்டும்
വിടവാട്ടോ എന്നാറാളുവാ എനിക്ക് സീനാണ് അറ്റൻഡൻസ് വേണം. ഹുസൈനുദ്ദീനാളിലേക്ക് ചോദിക്കുന്നുണ്ട് സാർ ഏവി ചോദിക്കുന്നുണ്ട് അറ്റൻഡൻസിനാണ്. ഓരോ അറ്റൻഡൻസ് ജൽദി ആക്കുക. अटेंडेंस करना आ जाए सभी सब
அன்மியூட் ஆயிருக்கடா. நிச்சயமா அன்மியூட் ஆயிருக்கடா.
International Conference on Environment and Health. In our midst, today's chief guest, Sri K. Justice Mohamed, who is here to enlighten with us in the topic of biodiversity conservation in India and the relevance to environment and health.
In the introduction, I need to say in the inaugural section, you heard many things about his biodata, his profile, his academic excellence. Here to highlight few of his academic excellence, which need to be inform you, particularly as a student. Sir is a personality to be imbued, to be followed, to be have you in your life for your development. Hence, first of all, I thank Dr. Justin Mohan sir for accepting our invitation to be here. Actually, you people are very much fortunate because sir recently he has transferred, I need to say, his completed duration as a secretary of biodiversity of India, that is MBA. Many people inquire what is formerly, then what is now. Hence, I am here to inform you that technical part. Recently, sir has completed duration as a secretary for National Biodiversity of India, Government of India, and is going to take over a new assignment from March 10th as the principal chief conservator of Forest, Government of Kerala. In the meantime, for 10 days or 15 days duration is idle. Hence, we are using, but I am using it because once he joins, it is not possible for him to spare his valuable time with us. And on behalf of Jamal and the Department of Zoology, I profusely thank for accepting the invitation and coming over here. Thank you, sir. So today, I is my senior and good friend, family friend. And what he is today is out of sheer hard work. And after his graduation, just a year, when he completed his post graduation in the second year itself, second year of MSc, he got service. These are the things you people, you students have to note, hence I am insisting this. When he, like our Siva Kumar, that is who completed NET examination. In our list, we have one student who completed the second year, now he completed NET examination. Likewise, sir is in the second year, first year itself, after because for writing UPSC exam, third, that is graduation year, and the first MSc he wrote the exam with the graduation degree, and while completing in the second year itself, we got the result, we got the service of Indian Forest Service. Okay, so after that, he was moving further to go for IAS examination. He has a time, age, everything. Without joining that service, IFL service, in the second year, he started again. I like to highlight that our DGP, Tiruchirappalli, Rainbow, all are the seniors of that institution. So those persons are the inspiration for the team who is studying, particularly civil service examination in the agriculture college point. Okay, so after in the second year, after second year, he wrote for, he set for another examination UPSC, and that time he cleared IPS. Again he cleared IPS, and twice he cleared IPS in the cadre of Kashmir as well as Maharashtra. In the meantime, he wrote many exams like Group 1 services. He got DSP, Group 1 services, Deputy Collector, like that. And he did not join that service, and later on after various discussion, he joined in the Forest Service. After 3 years of attempting a different examination, he joined the service which he cleared during time of student life. Okay, again he joined the service. So from that day, his academic as well as administrative career begins, and he headed, as our introducers are pointed out, his biodata. He headed many various projects with his above our head. Many crores of projects he completed. He completed a pet project of our former Chief Minister of Kerala. That is RFP project, as far as my some knowledge is concerned. He has been High Commissioner Secretary in Sri Lanka, rehabilitation work. So many landmark activity he did, and presently he is going to join as a President Conservator of Forest. He is blessed with 2 children, 1 daughter and boy. And so recently, last month, his daughter joined and the medical service. Sorry, medical MBBS course. So God has blessed him. And with this brief introduction, I am very much happy to welcome Sri Justin Mohan to dissipate the information related to conservation. Thank you, sir. respected now they state the professors and students who are attending this conference. So I have been asked to give this technical talk on biodiversity conservation and its relevance to environment and health. So that is the topic of this book. It's a very vast subject. It's a very vast subject. It cannot be covered within an hour, but I'll try my level best to bring in some important information so that it will be useful for you because you are all in biology background. Most of you are from zoology and botany. So, zoology and botany, as you know, are the core biology. And particularly zoology, because you can fit it into any other sector, whether it is medical or veterinary or health, environment, forest. You can fit it into any sector because that is the opportunity and scope for graduates passing from zoology. So, if you can pay attention to this lecture for the next 40 minutes, I try to cover within that. So, I'm sure that it is going to help you in the small way. So, I always begin my presentation with a null hypothesis, thinking that the audience may not be knowing about the subject. Some of you may be knowing, but that null hypothesis gives me a motivation to tell whatever I. So I presume that for the next 45 minutes, if you pay attention, you will know something which you do not know till. So with that assumption, I begin this topic. Next. So this is the plan of the presentation. We are going to see what is biodiversity, what is its importance, why we need to conserve our biodiversity. As you know, when we talk about environment, there are two major components. One is the living part, that is the biotic part, and one is the non-biotic, that is the non-living. So biodiversity comes under the biotic part that has life. The component which has life and the component which does not have life, like soil, they together forms the environment. So we are going to focus on how biodiversity is going to have an impact on environment. We are also going to backtrack. We are also going to see the Indian scenario, what is happening in India, and also the global scenario, what is happening to the globe at the global level, and what is the regulatory framework, what are the major laws to conserve our biodiversity. I will also touch upon what is the Biological Diversity Act and also the major challenges which this sector, environment sector, is facing today and the way forward. And finally, we'll have a small interaction. We'll close the session by 1 o'clock. Okay? 1 o'clock, the lunch will be ready, so we comfortably close the session by 1 o'clock. In the interaction, you can always ask me the questions. I will speak bilingual because some of the participants are from other states who are attending this program. So I will have a bilingual type of presentation. But if you have any doubt in between, also you please stop. So the more the doubts are, more interaction. Whatever doubt you are having, the person next to you also will be having, but he will not be willing to open his mouth. So if you ask, it is going to benefit the entire audience. Okay. Next. So what is biodiversity? Biodiversity is every species on earth which has life in it, that constitutes biodiversity. The variety and variability of all the life forms that is found on earth. Since you are all zoology students, so you are handling only biodiversity. You are going to handle biodiversity in graduation or post graduation, and if you are choosing a research sector, generally you are going to handled by us. So the variability and variety of all life forms on Earth constitute diversity. And there is diversity in this biology. Biological forms have a lot of diversity at the genetic level, at the species level. Say for example, like mango, you have n number of species. About 5,000 species are there in mango. That is a species biology. Mango is a species. Like paddy is a species. There are more than 1 lakh varieties of paddy. So that is a diversity. So there is a species diversity. Then there is the ecosystem diversity. So every ecosystem has a diversity. What animal you find in a desert ecosystem cannot be found there. Like you are seeing camel in Rajasthan, but you don't see camels only in the zoos. So there is an ecosystem diversity. Similarly, diversity is also found in terrestrial and aquatic. What you find in oceans cannot be found in terrestrial. Terrestrial, we are all living in terrestrial, but oceans have the maximum diversity. So diversity is either in the terrestrial form or in the biodiversity has direct relevance for whatever we eat. Is the biodiversity and also has relevance for water security. So most of our water security is coming from the forest, which is the core component of terrestrial life. So the forests provide the required water because they control the hydrological cycle. And also they have relevance in food production, for agriculture, health, because all the antibiotics which you are having, which you are taking, is all from one species. Can anyone tell what is the major species of biodiversity that provide antibiotics for use? Anyone? It's a microorganism.
Fungus.
Anyone?
Fungus.
Fungus provides only 5%. Major, of course, fungus was the first microorganism that produced antibiotic in the form of penicillin, Penicillium rotatum. But subsequently, more than fungus, 95% of the antibiotics today are produced from a species called bacteria. What type of that? Not equal. It's called streptomyces. So whenever there is a slight rain, there is a unwashed, there is no more smell over there. Actinomyces. Actinomyces. It's an actinomyces. The form of bacteria. It's an actinomyces species. It's called streptomyces. All antibiotic, whatever erythromycin, aminoxidil, fever, cold, cough, it's all coming from streptomyces. Okay? So that is a biological. So it has relevance for our health sector, right? And also for the industries, food industry. So for example, yeast is a fungus, so it plays a major role in the food industry. Mushroom, single cell algae like spirulina. So microorganisms also have a lot of relevance to food industry. See, whatever I am telling you, if you can take some points, going to help you in some interview. You are going and sitting before an interview board. Some basic questions they will ask. So it will help you at the time of interview when you are approaching any interview. Okay. Biodiversity also has a lot of indirect. It has a lot of indirect ecosystem services. As I told you, forests control the hydrological cycle and also produces the carbon cycle. See, you must have heard about greenhouse gases. So the major component of greenhouse gases is carbon dioxide. Carbon dioxide is a greenhouse gas. So trees and the forest ecosystems actually help to absorb the carbon dioxide for photosynthesis. Photosynthesis, carbon dioxide is absorbed. So oxygen. So it helps in carbon sequestration. Excess carbon dioxide or what happens? It increases the temperature of the earth. So that is called global warming. So major greenhouse gas apart from methane, this is the major greenhouse gas. So carbon sequestration, biodiversity. Similarly, pollination. You must have studied honeybees. Apiculture, we subject to the graduation. So it not only honeybees, lot of other insects, bumblebee, other insects, they help with pollination. Seed dispersal, birds are helpful in seed dispersal. So they provide lot of indirect ecosystems. They have nutrient recycling, soil productivity, nutrients recycling, microorganisms, major contribution from microorganisms for nitrogen fixation. Microorganisms, bacteria, atmosphere nitrogen and air fixed to the soil. So from soil, the plants use it for that. Okay. Similarly, certain elements cannot be soluble as. But say for example, phosphorus. Yeah, other than the plant is soluble form the nitrogen. So microorganisms help in solubilizing phosphorus. Plant in the weather and the solubilize. You will be surprised to know, and near that, there won't be any water, complete drought, but still some plant will be green. Dry season, how it happens? Outside near peak of May, June, there won't be any rain, but still there will be plant having leaves, and from there they get the water. That is because of the fungi called mycorrhiza. That is the God's gift for plants during drought season. It has the capacity. One soil is a water molecule. And the water molecule root absorb. Roots cannot absorb that water that is around the soil molecule. It is only this mycorrhiza called BAF, particular organism. That is the capacity. And the fungi has the capacity to absorb that water and give it to the plant. Another deciduous trees on the non-deciduous trees also have to survive. So biodiversity contributes a big way for directly. Next. But what happens is of late, there has been extinction of species. People say the last 100 years, there has been 100 times increase in rate of extinction. Okay. Last 100 years left to Bali. What has happened? Because extinction, there are two ways by which a species gets extinct. Extinction once for all is bird. Earthland once for all bird. One is in the natural form of evolution, and the species bird. Okay. Evolution, that is part of evolution. Whichever species survives, Charles Darwin's theory 80%. Survival of the fittest, whichever species survives, that will be remaining on other species will die. That is one form of extinction. But artificially man-made extinction, whenever you are going in for large scale development, destroying the forest, okay, hunting, excess pollution, these are the man-made forms of destroying an environment leading to extinction of. That was the last 100 years. Researchers informed that about 100 times there is a loss of species in the last 100 years. Next. Okay. Now we are moving to why we should protect our biodiversity. What is the reason? One is interdependence of species. Every species is dependent on another species policy. One species, if we lead to extinction, affects a series of other species, which is dependent on it. Preservation of life forms in a lot of wild genes available in forests. The recent biotechnology concepts of genetic engineering, where you can always isolate a very desirable gene found in the wild and transfer it to a cultivated species to increase production, to bring in pest resistance, disease resistance, salinity resistance. So, the species, the genes are not preserved. Future is going to help us. As I told you earlier, forests produce give lot of ecosystem services. Whatever air we are breathing, whatever water we are getting, either in the form of groundwater or in the rain, it's all controlled by the forest. They control the hydrological system. A lot of knowledge associated with biodiversity is also available, which has been preserved over centuries by tribal communities, rural communities, villages. The Paramariya arivu it will be linked to a particular biological process, a particular flower, linked. So it is also in our interest to protect those. This knowledge. Then as a humans, we have an ethical and moral, a right as also an obligation to protect all the other species. So, moral right. Other species, every species in this earth should live peacefully in their own. And of course, we also know that biodiversity is a source of livelihood for farmers, fishermen, tribal communities who are living in the forest. Livelihood, biodiversity. Flower, medicinal plants, and forest land. Collect money. Nearest tribal cooperative society. They will earn their livelihood. So it's a source of livelihood and for our life. For humans, for all species. They all depend on another biodiversity for them. And also, of late, the last 100 years of Earth, there is a lot of biodiversity that was happening. Biological species, traditional knowledge, steal money, they will make a lot of money for patent and the knowledge and the product and the process. They will patent it. They will be making a lot of money. It's called biopics. Okay, other check fund. So it's a major issue in biodiversity. Next. So coming to the Indian context, the total geographical area in the world, there are about 200 odd countries. Recently, December 1st, I attended the 3D conference in Montreal. It's called Conference of Parties on Conventional Biological Rights. Lady population street level population population 5 population 18% of the global population. Almost 1/6. Area only 2.4%, but our population contribution to the world is 18%. 1 in 6% in the world is 8% of the globally recorded species in India. 8%, about 156,000 species are there in India, recorded by Zoological Survey of India and Botanical Survey. They are the two highest scientific bodies of the Ministry of Environment and Forest, which are mandated to record all the species. Zoological Survey of India, Botanical Survey. Our website level information system. There is a recorded species of 156,000. That is 8% of it. Globally recorded species. Ours is a mega biodiversity. One of the 16 mega biodiversity. Biodiversity map, there is an equator. There is a Tropic of Capricorn, Tropic of Cancer. Most of the biodiversity is between the Tropic of Cancer and Tropic. So in the Nadala there will be biodiverse rich countries. Countries away from the Tropic of Cancer and Tropic of Capricorn will be economically rich countries. Biodiversity ரொம்ப poorer. காலகாலமா என்ன நடக்குது? And the economically rich Western countries have always thrived on the biodiversity in the countries between the Tropic of Cancer. Okay, so India is one of the 16 mega biodiverse countries. நமக்கு ஒரு 36 hotspot 36 hotspot ல நம்ம நாட்டுல மாத்திரம் 4 hotspot. There is a lot of wild endemic species. Livestock 5.6% of the world's GDP நம்மளோட நாட்டுல. And 18% of the livestock population also. Yes, நம்மளோட world over 18% of the livestock. Next. So this is the 10 broad biogeographic zones available in India. As I told you earlier, what is found in one biogeographic zone cannot be found in another. Okay. So these are the 10. So India is a biodiverse countries with biogeographic zones. The presentation will be here. You can always take it. Next. So, over the years, there has been a lot of developments, particularly on the colonization. Last when the sea route to various countries were found out by Vasco da Gama and the Portuguese. People from Europe started occupying different parts of it. Africa, South America, Portuguese and Spain அதனாலதான் பாத்தீங்கன்னா அங்க எல்லாம் ஃபுட்பால் கண்ட்ரிஸா இருப்பாங்க. அதனாலதான் அங்க விளையாடுற போர்ச்சுகல் சொல்லிட்டு இந்த மாதிரி பிரேசில், அர்ஜென்டினா எல்லாமே போர்ச்சுகீஸ் பேசும். போர்ச்சுகீஸ், ஸ்பெயின் எல்லாம் சவுத் அமெரிக்கன், அவங்க ஒரே பாஷை. இது பாத்தீங்கன்னா ஆப்பிரிக்கா பாத்தீங்கன்னா இங்கிலீஷ் அண்ட் பிரெஞ்சு. Asia predominantly English. They came here to explore this area and finally settle here. They found that okay, the area was super. Various species, they started settling. They wanted to settle. They wanted to use this country to exploit. So, on the process, what has happened? Destruction of forests. One side. No side, a lot of advances in science and technology, particularly in the medicine. So medical field, advances for 100 years back, diabetes people used to die at 40-45 years. Only there, diabetes. But now we know what is diabetes, avoid from array of medicine. Similarly, cancer. Few years back, only there, cancer. People know advanced medical. Over a period of time, lifespan started increasing. 40-45 வயசு இருக்கிற பாப்புலேஷன் ஸ்லோவா 70 to 80 years. When we got independence 1947, our population was 33 crores. 33 கோடி தான். இன்னைக்கு actually 2021 ல எடுத்துறோம் census. But because of COVID we did not take. It is expected that we would cross 1.4 crores. Those are not right. அப்போ 4 மடங்கு ஏறிச்சு பாப்புலேஷன். 33 கோடி இல்ல 140 கோடி. ஓகே. சோ இந்த இவ்வளவு மக்களுக்கு சாப்பாடு கொடுக்கணும். Particularly countries like India, we have to take care of the growing population. After we got independence in 1946, successive governments focused on food production. Allah highly productive forests எல்லாம் வந்து destroy பண்ணாங்க. India மாத்திரம் பல நாடுகள் ஏன்னா we all got independence at the same time. At the World War II அப்புறம் தான் பல நாடுகள்ல இந்தியா வெளியே. So at the time the population take care of it. They started producing more. The easiest way was to destroy a forest along the agricultural land. Okay. So slowly it became intensive agriculture. நிறைய pesticides, chemicals போட்டு they wanted to increase the food production. Take care of all. ஏன்னா geographical area அப்படியே தான் இருக்கும். For example, இந்தியாவோட geographical area பாத்தீங்கன்னா 329 million square kilometers. அதை நம்ம எக்ஸ்பேண்ட் பண்ண முடியாது. But we have to take care of the population which has expanded 3.5 times, 4.5 times. So as the intensive cultivation we have to go in for more pesticide, chemicals. So a lot of developments have happened in the last 6 years. So not only specific to India, it has been happening across the world. So the world community for the first time, under the leadership of our United Nations, came together at Stockholm, Sweden. Other than 130 countries are participating. India participated. At the time, our Prime Minister, our Srimati Indira Gandhi, attended. On 5th June, every year we are celebrating World Environment Day on 5th June. So the countries come together, telling that enough is enough. Our environment, it is not restricted to one country. What if India is not taking care of its environment? It is also going to affect other countries, not only neighboring countries, countries foreign. So in Stockholm, they take a decision that we have to take all the countries who are attending this conference will take suitable measures to protect their environment. Okay. So, at the conference, the United Nations Environment Program was Guru of this program at the time. Okay. Next. Appo Indira Gandhi attend பண்ணிட்டு வந்தாங்க. வந்ததுக்கு அப்புறம் she had a 3-pronged approach. 3 ways she is trying to address this commitment given at the Stockholm. One என்ன பண்றாங்க? She is amending the Constitution of India, which is the supreme law of our country. So they are bringing in 2 new articles. It is called right to life, right to fundamental duty, and directive principles of state. Yeah. Under Article 48A, the state is mandated to protect the environment. It is the responsibility of the government, both central government and state government. Other than under 51A, the fundamental duty. They are telling that all citizens, it shall be the fundamental duty of every citizen of India to take care of our environment. On the Constitution amendment, the Stockholm Conference. Yeah. Next. Then she brings a lot of laws. The Water Act 1974 to take care of the water. Environment is also the water and the moon is the environment. Water and correct the moon Water Act in 1974 to prevent and control pollution 1981. And then she brings, of course, she died in 1984. But his son, Rajan, came to power and brought in 1986 the environment protection. So, air is separate law, water is separate law, environment is separate, land is separate. For comprehensive legal protection to take care of that. Okay. Then the government thought that apart from this, we also have to take care of our forest. Because forest is actually the crux of biodiversity and other. So they enact the wildlife protection. This wildlife is very critical for the health of the forest people. Okay, no? Wildlife is not forest survival. Wildlife is not, it will become a desert. Okay. And then to prevent diverting forest land for non-forest produce, non-forest activities. Okay, the Forest Conservation Act was enacted in 1980. So, that's the procedure. The procedure follow பண்ணலனா minister இருந்தாலும் நீ ஜெயிலுக்கு அப்படி ஒரு சட்டம். Forest Conservation Act, and Wildlife Protection Act was also enacted. They are now subsequent to the Stockholm Conference. Next. This is government brings a lot of policies, new projects. They are setting up national parks, wildlife sanctuaries, biodiversity reserves. A lot of zoos, botanical gardens, herbariums to store genetic material. Okay, gene banks. So both in situ conservation in the form of setting up national parks, sanctuaries, biosphere reserves, and also ex situ conservation in the form of botanical gardens, zoos, gene banks. So in the very comprehensive, they want to protect this environment. These are the policy decisions of the government subsequent to the Stockholm Conference. Next. Now, as this was going on, now the Western countries started stealing a lot of biological resources from biodiversity rich countries, and the traditional knowledge which is available in these countries started patenting outside. Okay, India is ஒரு particular disease say for example English medicine la cure இல்ல, but ஒரு tribal settlement la இருக்கு அந்த cure வச்சிருக்காங்க, ஒரு ஆதிவாசி family இருக்கு, அதை திருடிட்டு போய் அங்க பேட்டன்ட் தெரியாது. கிளாசிக் கேஸ் இதுல டர்மரிக் கேஸ், நீம், இதெல்லாம் அந்த காலத்துல இருந்தே நம்ம நாட்டுல ஃபாலோ பண்ணுங்க, அங்க போய் யுஎஸ்ல போய் பேட்டன்ட் இருக்கு. So there has been lot of biopiracy, biological piracy. அப்ப என்ன பண்றாங்கன்னா, in 1992 there was an Earth Summit, World Summit. It changed the face of environment protection. It happened in Brazil, Rio, Earth Summit. அதுல என்ன பண்றாங்கன்னா, environment மாத்திரம் பார்த்துக்கிட்டா பத்தாது, we have to take care of the biodiversity also. அப்ப என்ன பண்றாங்கன்னா, 3 convention create பண்ணு, 3 conventions, 3 bodies they are creating under UNE. ஒன்னு வந்து convention to combat desertification. What land is there, you make it productive. Desertified areas on the productive ecosystems. அதுக்கு ஒரு convention, and the convention headquarters on Germany. Second convention, convention on biological diversity. Okay, அது Montreal, Canada. Once on the desertified areas on the productive areas of Montreal, the biodiversity market. அதுக்கு ஒரு ஆஃபீஸ். மூணாவது கன்வென்ஷன் வந்து UN Framework Convention on Climate Change. அது பாண்ட்ல தான். இப்போ ரீசெண்டா வந்து ஈஜிப்ட்ல நடந்தது அதோட கான்ஃபரன்ஸ் in November, climate change conference. எல்லா வருஷமும் நம்ம பிரைம் மினிஸ்டர் போற வருஷம் அவர் என்வரன்மெண்ட் மினிஸ்டர். அதே மாதிரி that happened in Egypt, Sharm el-Sheikh. ரெண்டாவது biodiversity conference happened in Canada, Montreal. So இந்த மூணு கான்ஃபரன்ஸும் மூணு கன்வென்ஷனும் கிரியேட் பண்ணாங்க. மூணுலயுமே இந்தியா பார்க்கும். அண்ட் இந்தியா அந்த கான்ஃபரன்ஸ்ல என்ன சொல்றாங்கன்னா, இந்தியா along with all the other 196 countries has to enact law. அப்படிதான் biological diversity act அப்படின்னு இந்தியால ஒரு சட்டம் இருக்கு to protect all the biological species. Okay, 1990 2002 ல ஆக்ட் சட்டம் கொண்டு நெக்ஸ்ட் அந்த ஆக்ட்ல என்னன்னா, அந்தந்த நாடு இந்தியா அவங்க நாட்டுல உள்ள ஸ்பீசிஸ் அவங்களுக்கு தான் சொந்தம். ஒண்ணு, அந்தந்த நாட்டு must take care of the conservation of the species to ensure sustainable availability of the species. என்ன வந்து புது புதுக்கா இருக்கு என்ன சொல்றாங்க அப்படின்னா, the companies எல்லாம் இருக்கு, pharmaceutical companies, fragrance companies, அவங்க வந்து biological resources from forest ஆனா முன்னாடி எக்ஸ்ட்ராக்ட் பண்ணுது. They are making huge profit. அந்த ஆதிவாசி ஃபேமிலிக்கோ அல்லது அந்த விவசாயிக்கோ he does not know the actual value of the raw material. அவன் கொடுத்த raw material actual value. இவனுக்கு 1000 டைம்ஸ் அவன் வந்து ப்ராஃபிட் பண்ணாங்க. ஸோ இந்த கன்வென்ஷன் என்ன சொல்றாங்கன்னா when you are making a profit, நீ யாருக்காவது நீ ரா மெட்டீரியல் வாங்குறீங்க அவங்களுக்கு ஒரு போர்ஷன் ஆஃப் த ப்ராஃபிட் இன்க்ரீஸ் ஷேர். அதான் ப்ராக்டிஸ் ஆஃப் பயாலஜிக்கல் டைவர்சிட்டி. அதனால அவன் என்ன சொல்றான்னா யூ ஹேவ் டு டேக் த ப்ரையர் அப்ரூவல் ஃப்ரம் த நேஷனல் பயோடைவர்சிட்டி அத்தாரிட்டி. ஏதாவது ஒரு கம்பெனியோ ஒரு இண்டிவிஜுவலோ ஒரு பயாலஜிக்கல் ரிசோர்ஸ் ஃபார் கமர்ஷியல் மோட்டிவ் ப்ராஃபிட் மைண்டோட நீ வந்து அக்சஸ் அப்ரூவல் இல்லாம நாங்க எடுத்தீங்கன்னா அது வந்து அஃபென்ஸ் 5 வருஷம் வரை ஜெயில போகும். அதான் பயாலஜிக்கல் டைவர்சிட்டி. ஸோ அப்ரூவல் கொடுக்கும்போது யூ ஹேவ் டு கிவ் அ கமிட்மெண்ட் தட் அந்த எண்ட் ஆஃப் எவ்ரி ஃபினான்ஷியல் இயர் நான் இவ்வளவு அமௌண்ட் எங்கேருந்து நான் எடுக்கிறேனோ அவங்களுக்கு நான் கொடுத்தேன். இப்போ as on date it is up to 0.5% of the gains. 0.5% வரை அவங்களோட லாபத்துல இருந்து அவங்க வந்து ஷேர் பண்ணுவாங்க. ஓகே, நெக்ஸ்ட். ஸோ பயாலஜிக்கல் டைவர்சிட்டி ஆக்ட்ல பாத்தீங்கன்னா எங்க நம்ம ஜுவாலஜி ஸ்டூடெண்ட்ஸ் எப்படி ஆக்ட்ல வந்து நீங்க எப்படி கான்ட்ரிபியூட் அப்படின்னு. ஸோ ஸ்பெஷல் ப்ரொவிஷன்ஸ் இருக்கு இந்த ஆக்ட்ல. லைக் one provision is the government can declare a particular area rich in biodiversity as a biodiversity heritage site under section 37. அப்ப சப்போஸ் நீங்க நாளைக்கு அங்க வந்து ஒரு பர்டிகுலர் ஏரியா வந்து நல்ல பயோடைவர்சிட்டி நிறைய காய்கறிகள் வருது, நிறைய மெடிசனல் பிளான்ட்ஸ் இருக்கு, அல்லது நிறைய பட்டர்ஃபிளைஸ் இருக்கு அப்படின்னா அது வந்து பயோடைவர்சிட்டி ஹெரிடேஜ் சைட்டா நீங்க அங்க உள்ள எல்லா பஞ்சாயத்துலயும் ஒரு பயோடைவர்சிட்டி மேனேஜ்மெண்ட் கமிட்டி ஃபார்ம் பண்ணிருக்கு. இந்தியாவில் பாத்தீங்கன்னா 2,77,000 பயோடைவர்சிட்டி மேனேஜ்மெண்ட் கமிட்டிஸ் ஃபார்ம் பண்ணிருக்கு. அந்த கமிட்டி வழியா நீங்க கவர்மெண்ட்டுக்கு ஒரு ரெக்கமெண்டேஷன் அவங்க முடிவு அதை வந்து பயோடைவர்சிட்டி ஹெரிடேஜ் சைட்டா. அதே மாதிரி எல்லா கிராமத்திலையும் அந்த பயோடைவர்சிட்டி மேனேஜ்மெண்ட் கமிட்டி ஹாஸ் டு பிரிப்பேர் a register of biodiversity in that area. It's called people's biodiversity register. அதுல பொதுவா அந்த மக்கள் அந்த டிஎம்சி மெம்பர்ஸ் இருக்காங்க, பெரிய அளவுக்கு அவங்களுக்கு வந்து அந்த ஐடியா இருக்கு. அதுல வந்து நம்ம பாட்டனி ஜுவாலஜிஸ்ட் அவங்களுக்கு ஜுவாலஜிக்கல் நேம் தெரியாது, பொட்டானிக்கல் நேம் தெரியாது, என்ன யூஸ்னு தெரியாது. பட் அதுக்கு தட் வில் ஹேவ் சம் கமர்ஷியல் நேம். ஃபார் எக்ஸாம்பிள் லேக் ஒரு ஏரியால இருக்க லேக் இருக்கு, அதுல வந்து நம்ம வந்து அந்த ரெஜிஸ்டர்ல கொண்டு வரணும். அதே மாதிரி ட்ரெடிஷனல் நாலேஜ் ஒரு பர்டிகுலர் ஏரியா. ஸோ நீங்க ஏதோ ஒரு ஊர்ல நீங்க ஒர்க் பண்றீங்க, யூ கேன் கோ அண்ட் மீட் த பிஎம்சி மெம்பர்ஸ். So you can go and talk to them. அவங்க ஆல்ரெடி ப்ரிப்பேர் பண்ணி வச்சிருந்தாங்க. You can try to update them, help them. இதெல்லாம் நீங்க தப்பா எழுதிருக்கீங்க, இதெல்லாம் மாத்தி. So zoology students can also contribute in biodiversity by updating the people's biodiversity. And also இந்த ஆக்டோட சிறப்பை பத்தி அவங்களுக்கு தெரியப்படுத்தும். அவங்களோட கடமைகள், டியூட்டிஸ், ரெஸ்பான்சிபிலிட்டிஸ். As a graduate coming out from the zoology or botany department, you should be aware about what is the role of the BMC in protecting them. Okay, இந்த சட்ட பிரகாரம் ஒரு கமர்ஷியல் மோட்டிவோட ஒரு இண்டிவிஜுவலா போய் ஒரு பயாலஜிக்கல் ரிசோர்ஸ் இந்த மைக்ரோ ஆர்கானிசம், இன்செக்ட், பேர்ட்ஸ், அனிமல்ஸ் எடுத்தாங்கன்னா அப்ரூவல் இல்லாம எடுத்தாங்கன்னா penalties, 5 years imprisonment or 10 lakhs fine or proportionate to the loss caused to the biodiversity. Okay, next. So இந்த சட்ட பிரகாரம் biological resources if they are access for commercial utilization or for research or for taking IPM patent. இந்த மூணு ஆக்டிவிட்டி predominantly இந்த மூணு ஆக்டிவிட்டி you have to take the approval from the national biodiversity authority or to the from the state biodiversity. Okay, research by Indians is excellent. நீங்க இப்ப சப்போஸ் நீங்க ஒரு ஜமால் மொஹமெட் காலேஜ்ல you are undertaking research அப்படினா அதுக்கு எந்த அப்ரூவல் வேணும்? But if you are a multinational company or a foreign linked company, then you have to take that. But India number institution or number university on your own, you are undertaking business code approval. But if you want to hand over the business result to a multinational company, section 3(2) company, then you have to take the approach. National biodiversity பத்தி அப்படி அப்ரூவல் இல்லாம நீங்க கொடுத்தீங்கன்னா ரெண்டு உங்களுக்கு வர வேண்டிய லாபம் அந்த கம்பெனில இருந்து வர வேண்டிய லாபம் போச்சு. ரெண்டாவது அப்படி கொடுத்தீங்கன்னா it amounts to an offense. The concerned persons எல்லாம் கொடுத்துருவாங்க. குடிக்கலாம். ஓகே. Next. So அந்த பணம் ஒரு அந்த benefit sharing that is being shared by the company, அது 5% will be taken by the NBA, National Biodiversity Authority, or State Biodiversity Board, and balance 95% will go to the communities. In the village, our resource okay. So, in the amount they can use it for the cause of conservation or for promoting or cultivating or propagating or safeguarding a particular resource that has commercial value. So, health sector, the conference. ICEH is the face of India as on date. ICEH companies are going. For 10,000 ICEH companies. Yeah. So they have been continuously taking biological resources from the wild. So 85% of the raw material comes from the forest, 15% from the cultivated species from rural areas. Farmers are their own land network. So in the species of cultivator or protect for the amount use for it. And that part but நேரா அந்த எந்த வில்லேஜ்ல பார்த்தா நேரா உங்களுக்கு ட்ரெடிஷனல் நாலேஜ் நம்ம இந்த ஃபேமிலில இருந்தா அந்த ட்ரெடிஷனல் நாலேஜ் வந்தது அப்படின்னா அந்த ஃபேமிலி இல்லைன்னா அந்த பிஎம்சி அந்த பஞ்சாயத்துக்குள்ள ஃபங்க்ஷன் வந்து பயோடைவர்சிட்டி மேனேஜ்மென்ட் அதை அவங்க யூஸ் பண்ணிக்கலாம் ஃபார் கன்சர்விங் த ஸ்பீசிஸ் ஆர் ஃபார் எனி சோஷியோ எக்கனாமிக் டெவலப்மெண்ட். அந்த ஃபிளெக்சிபிலிட்டி கொடுத்துருக்காங்க ஆக்ட்ல. அந்த கிராமத்துக்கு ஏதாவது தேவை இருந்ததுன்னு வச்சுக்கலாம், ஒரு பப்ளிக் டாய்லெட் கட்டணும், ஒரு ரோடு ரிப்பேர் பண்ணணும், primary health center வந்து furnish பண்ணுவோம், tribal school furnish பண்ணுவோம் அப்படின்னா அதுக்கு யூஸ் பண்ணுவோம். Okay, next. So coming to the major issues pertaining to biodiversity, as the morning another speech highlight global warming. Tremendous global warming that is happening here. January it is raining in Chennai. Can you believe? India மழை பெய்யுது. It is not the time for it. Today floods, unbelievable floods. Today drought. There is whether we realize it or not, global warming is happening. Yeah, the global warming is because of higher production of greenhouse gas on one side. Other side, and the carbon dioxide absorb பண்ற trees குறைஞ்சிட்டே இருக்கு. ஓகேங்களா? இதனால என்ன ப்ராப்ளம் ஃபார் தி என்வாயரன்மெண்ட் ஆர் ஃபார் தி ஹெல்த் அப்படின்னா ஒரு நல்ல ஒரு மாய்ஸ்ட் என்வாயரன்மெண்ட் திடீர்னு டவுட் ஆச்சு. certain species will be living there. அது என்ன பண்ணும்? It will move to another species. For example, some microorganism is living there. It needs a host. அது இன்னொரு இடத்துக்கு போயிடும். அது தெரியாம மனுஷன் சாப்பிட்டாங்கன்னா அது அப்படியே கோவிட் தான் இருக்கும். ஓகே. Lot of zoonotic disease. உங்களுக்கு ஆல்ரெடி தெரியும். dengue fever எல்லாமே zoonotic disease. Plague ஏதோ ஒரு zoological சேர்ந்த ஒரு ஸ்பீசிஸ்ல இருந்து வரது நிறைய disease humans affect. So many diseases come from insects or from contaminated water. So these are all health issues when we are not taking care of them. Lot of chemicals are dumped in the agricultural ecosystem. That is leading to pollution. Another அந்த காலத்துல வாழ்ந்த மக்கள் வந்து ரொம்ப லாங்கிவிட்டி சில பேர்லாம் நல்லா வேணா நல்ல ஹெல்த்தி லைஃப் பொல்யூஷன் ஃப்ரீ லைஃப். இப்போ வந்து அந்த அளவுக்கு இல்லை. வாட் எவர் வி ஆர் ஈட்டிங் cannot be declared as pollution. ஏன்னா chemicals are dumped, pesticide, herbicide, fungicide, okay, fertilizers. So it will knowingly and unknowingly get into the plants. Other numbers suffer. So a lot of pollution. And there is always a constant debate between development and environment. Between economic available ecology வேணும், development வேணுமா, environment வேணும். So in that process, the country has to grow a lot of connectivity வேணும், road போடணும், railways கொண்டு வரணும். அப்போ வந்து you are going encroaching into the forest. அப்போ அந்த man animal conflict நடக்கும். Animals will start moving into residential areas, causing loss to life and property. மனுஷன் போய் deer சாப்பிடணும்னு நினைப்பான், அல்லது ஒரு காட்டு பண்ணியை சாப்பிடணும்னு நினைப்பான், வயலுக்கு. Without knowing that every organism has a lot of microorganisms. எதுக்கு எப்படி கோவிட் வந்து? Because this people do not know that every organism, every microorganism is there in every living species. நம்ம உடம்புல lot of bacteria. இப்போ இன்னொரு ஸ்பீசிஸ் ஒரு வவாலோ அல்லது ஒரு பேங்கோலியனே போய் சாப்பிடும்போது அந்த ஸ்பீசிஸ் அந்த மைக்ரோஆர்கனிசம் உடம்புல வரும். It will take time to adjust into the human metabolism. அது முடியலும் what happens, that people die. Spanish flu அது மாதிரி தான், நம்ம கொரோனா அப்படிதான். When it takes time for that microorganisms to settle in the humans, okay பா இது நம்மளோட புது ஹோஸ்ட், இந்த மாதிரி இங்க தான் நம்ம வாழ்ந்து ஆகணும் அப்படின்னு அது period is very long. So when you are destroying forest ecosystems or during global warming, such harmful microorganisms move into other species, okay, and ultimately affect our humans. And you know that almost 65 lakhs people have died because of COVID. Here, as per estimate, about 5.5 lakh people have died. So, it's on the recorded one. Recorded 5.35 lakhs. India, second largest death in the world after the United States. United States, I think 11.5 lakhs died. We lost about 5.3 lakh people. Recorded one. So there has to be a balance between environment and development. More development at the cost of environment, more problems for the future. Next. So what we are going to do? We have to restore our ecosystems. One is the forest ecosystem. Lot of wildlife trade is happening. Under the myth that tiger bones சாப்பிட்டோம்னா நமக்கு இந்த benefits கிடைக்கும். So lot of myths are happening. So காலகாலமா நம்ம இந்தியாவை பொறுத்தவரைக்கும் wildlife has always been an integral part of our culture. அதனால தான் நம்ம வந்து we have revered our animals. There is a cobra or elephants. We think that their presence actually helps us. So we have to take care of our forest and wildlife. We have to restore it. Okay. And of course, there are a lot of global agreements. CITES is there now. I mentioned international trade and endangered species. Almost many of the biodiversity countries have their own wildlife laws. By and large, we have tried to prevent wildlife trade or wildlife poaching, but still a lot of things are happening. So it's very difficult. Wildlife kill it's very easy. It's a very porous border between forest and a residential area. The border is very porous. For puliya vechi unnowingly you eat it. The land is covered. Okay, next. Restoring our agriculture ecosystem should be our next objective. 2014 when this government, India government came, they started this control soil health card scheme. The objective of the scheme was, there is always a myth among the farmers that fertilizer dump yield. And the myth demolished for the scheme. Optimum application of fertilizers will help to preserve the soil health, because microorganisms are there. Not only preserving the soil health, it will also preserve the health of the crop, increase the yield, reduce the cost of cultivation. Salary portion, major expense for cultivation is fertilizers. So restoring our agricultural ecosystems. They also brought in this drip and sprinkler irrigation. Why is the exploitation of groundwater without knowing that the crop is there over current power free people used to extract. Now there are a lot of subsidy schemes for drip and sprinkler irrigation. So we have to restore the agricultural ecosystem. Yeah, beneficial microorganisms full of beneficial organisms will die. Biodiversity will go. Similarly, full of pollute chemicals over dump thinking that yield beneficial organisms. Okay. So we have to restore our agricultural ecosystem. Next. We also have to restore our aquatic ecosystem. Pollution. What we have seen, we have a lot of NGOs are working to take care of our Uyamata. They wanted to irrigate this part of the area. The canal. But of late, every city will have a drainage center. Actually, it was once a thriving river, which was useful for irrigation. Agriculture used for a river. Population the easiest way to dump our waste is to find a outlet. To make a heard about BOD, biological oxygen demand, chemical oxygen demand, graduation, so it increases, affecting the health of the aquatic ecosystem. And fishes and okay, there are zoonotic diseases or chances. So we have to take care of our water bodies. Being as zoology students, you should take up the lead in the work. Apart from your work, you should also devote a small contribution. That is your contribution to society. Small time for something like that to create awareness to take care of the environment in your locality. You can contribute to the well-being of the environment in your locality because you are technical people. What knowledge you have, the technical knowledge of a common manifestation. So you can help. Next, restoring our urban ecosystem. This is our contribution. City level, easiest way is to plant a tree. Land this is the major contribution. Because you already know that trees actually absorb carbon dioxide. Carbon sequestration is a very important component. Small contribution from your side to plant trees will help in a major way to protect our environment. Historical place. Tiruchirappalli. I worked for it. Area that area and the area of the Science University was there, created by LPT those days. War demolition housing project agriculture college or engineering college set up. University was the administrative capital of LPT. We planted these trees, I think 10 years back. That is our contribution. We have been creating that awareness among those people also in the that wherever possible, weekly or the that is our contribution to mitigate climate change. Yes. So thank you very much. If you have any, I told to stop by one. If you have any doubts, clarifications, most welcome. And for those who want to appear for any competitive exam, up to last you are most welcome. So only doubt.
That's my first.
Sir, so in this things are available. Is it the actually grown natural? Is it the right way of making that is not timber? Actually, she looks like a timber. Timber. Is it the right way of cultivating in the timber? This the part of the border of the class. We are displaying a forest for our own convenience. So road border was affecting railways. So forest was on the road. So, but handling of we have a particular way of landing people. Some civilians, number urban areas, number weak it's very costly. Forest timber calculation is a very costly. We cannot take care of timber like what we do in our kitchen. Tracker, undulating tracker. So, நம்ம வந்து நார்மலா ஒரு ஸ்பீசிஸ்க்கு ஒரு ஸ்பேசிங் வந்து ஒரு அந்த கட்டு இருக்கு இப்ப பர்టిక్యులர் அந்த சீட்லிங் ஸோ அந்த பர்టిక్యులர் ஸ்பேசிங் வச்சு ஆஃப்டர் 5 years we prune it or we thin it அந்த கட் பண்ணி சப்போஸ் 2/2 spacing 2/2 so that on the remaining source of trees will have better growth. So this is the one that this is. So I wanted to protect because these are all the development. That's it. That's all. We want to short timber. This much area is near for one timber. It's not growing extra large. And find the loop to penetrate deeply. Otherwise, we think. So when you think about if the soil is very rough, you cannot grow timber underside. So we have made artificial holes, then we have plants, then we get a big timber. So we have 1 more percent right now. So we are going to get 100 of these. What is the MOD for that? Please, immediately. What is the MOD for that? No, we are going to get.
I'm talking about land infrastructure. Sir, I think it's procedure. Suppose there is a development. So you are having that. Yes. 10 times more trees are planted.
Now we are going to 100 trees. 25 years before, we want to must have 100 trees. Suddenly,
development for you have to get the compensate state agency which is connected to plan
அதுக்கு land அவங்களே கொடுக்குது. ஆமா.
They have to plant and necessarily 20 years ago. So that's a technical. It's all over. Instead of
Nice, sir.
renewable energy. So that renewable source we must recycle or not. Innovating farmers.
Anything culture must be common method. Next to but Professor Jasmine has given a very lucid explanation of how there is a constant fight between environment and nature. And development must be in progress at the same time, but we think of the health of environment also. When there is a healthy environment, then man can also in every sense of it. My question is, on behalf of you, I am asking a question. Frequently, we see in the video, the animals like the elephants are improved, according to the habitations. And then we have the no man animal conflict வந்து அடிக்கடி நடக்கும். இது என்ன இதுதான் பீச் அப்படிங்கிறது நிறைய காரணம் இருக்கு. அது நான் சொல்றத விட சாதனை நல்லா
That is because when we encroach into a violent area, as I told you earlier, the option between ecology and economy. When there is a question between environment and development, when there is no other option but to encroach a forest, then you are road moving, you are going to resettle a tribal group of families in the forest. Actually, there is going to be a man-animal conflict. Man-animal conflict or violence. Predominantly, we are entering into their home. Out number we are going into their home and then blaming the animal. Because elephants for particular path, it's called elephant corridor. But we are blocking that corridor. Why did you? I don't know. And what many what you are doing is forest area, for example, the Coimbatore area. நம்ம போய் பெரிய ஆமா அவங்க என்ன பண்றாங்க இங்க காத்துல வீட்டுல சாப்பிடுறது கரும்பு இருக்குது கரும்பு அப்போ ஷாப்பிங் பேட்டர்ன் நம்ம மாற்றோம் அதை நம்ம அட்வொகேட் பண்றோம் இருந்தாலும் கேஷ் கிராப்னா ஃபார்மர் அப்போ you cannot blame the everybody. Once it knows that I want for illicit act அப்படிங்கும்போது எலிமெண்ட்ஸ் they enjoy having samples, it becomes out of control. ஒரு லிமிட்டுக்கு மேல போயிடுச்சு அது பாக்குறதெல்லாம் அதிகம். ஒன்ஸ் அதை ஃபேஸ் பண்ணிடுச்சுன்னா அந்த அந்த இடத்துல அது கிடைக்குது அப்படின்னா திருப்பி வரும். So it is more of a man-made problem in the form of development, destroying our forests, or in the form of cropping pattern, which the elephants want, or in the form of illicit arrangements. It is happening inside the forest or the outskirts of the forest, which actually attract. This is the main reason for start a conflict is an interface. So, I have walked from Valparai to Munnar. Valparai to Munnar. Or even the tribal settlement. It's called Eramala. This is a whole death to another elephant, full of elephants. Because the people living there and the elephants, they know each other. Not one man on the hot of the settlement. Elephant was in one place. He is not coming. I'm not the thing. So they have some sort of understanding. You know, number of hours. Every wildlife or forest ecosystem harmonious. They feel that tribals number of hours. But what happens is that when you are going and harming them, they will also area of. But then there is a conflict. There is the interface becomes negative. Otherwise, there is a people, tribal settlement, they only speak language. Fluent Tamil. The Pandya Raja, Solar Raja, attack the Pandya Raja, save put it by Munnar. And they found that it was super again settled. Raja and the war Raja the Mangal and get settled. So, but the life how to pure life. Not also eat only vegetarian. Muduan tribe அப்படினா புதுல குழந்தை வச்சிருக்காங்க. நீங்க கூகுள்ல போறீங்கன்னா தெரியும். உங்க தூய தமிழ்ல பேசுவாங்க. நம்மளோட லாங்குவேஜ் அவங்களுக்கு கஷ்டமா இருக்கு. இந்த இலங்கை தமிழ்ல பேசுவாங்க. அவங்களுக்கு ஒரு லாங்குவேஜ் நம்மளுக்கு போனோம்னா அவங்க நல்லா போகும். எந்த ப்ராப்ளமும் இல்லாம they are living very happily with the education. அதுதான் பயமா இருக்கு. They have a very wonderful understanding with the wildlife. இது வர அந்த எலிபென்ட் புலியோ அட்டாக் ஆகி ஒருத்தர் இறந்துட்டாரு அப்படின்னு நான் கேள்விப்பட்டது. ஸோ நம்மளா போய் என்கரேஜ் பண்ணி அவங்க கான்ஃபிளிக்ட் உண்டாகிற டைம்ல அந்த லாஸ் ஆஃப் லைஃப் அண்ட் ப்ராப்பர்ட்டி வருது. அதர்வைஸ் people live in harmony with the nature.
ICEH company 95% அதை பத்தி அந்த ஃபாரஸ்ட் ஏரியால இருக்கிற ரா மெட்டீரியல்ஸ் தான் யூஸ் பண்றாங்க. 85%. அப்ப அத நீங்க எதுவும் இப்ப குறைஞ்சிருக்காங்க. அந்த பயோபைரசி குறைஞ்சிருக்கு.
அது பயோபைரசி கிடையாது. பயோபைரசி கிடையாது. பயோபைரசினா
ஆக்சுவலா பயோபைரசினா ஒரு ட்ரெடிஷனல் ஃபேமிலியில இருக்கிற நாலேஜ் அவங்க எடுத்துக்கிறது பயோபைரசின்னு சொல்லியிருக்காங்க. அவங்களுக்கு அவங்க நாலேஜ் அவங்களோட வேலைக்கு நாலேஜ் ஒர்க் அவங்களுக்கு தெரியுது. இப்போ அவங்களுக்கு அது தெரியுதா, அவங்க இப்போ கொஞ்சம் இம்ப்ரூவ் ஆயிருக்காங்க அப்படின்றது.
ICEH companies they need raw material for their production. 7% இருக்கு. ஸோ 23, 25 மூலிகை பொருள் கலந்துதான் அந்த சிறப்பு பேஸ்ல விட்டாங்க. இவங்க எல்லாம் they have 3 sources. ஒன்னு வந்து அவங்களுக்கே லேண்ட் இருக்கும் கம்பெனிக்கு, இன்னொரு வந்து ஃபார்மர்ஸோட ஒரு கான்ட்ராக்ட் வச்சிருக்காங்க. Major supply is from the markets. Mandi, பெரிய மந்தி, டெல்லி, பாம்பே எல்லாம், or from the middlemen. அந்த மிடில்மேன் என்ன பண்ணுவாங்கன்னா அந்த ஃபாரஸ்ட்ல அவங்களுக்கு அந்த ரைட்ஸ் வந்து அவங்களோட லைவ்லிஹுட், அவங்களோட வாழ்வாதாரம், அங்க உள்ள வேற பூ, மெடிசல் பிளான்ஸ் எல்லாம் கரெக்ட். ஒரு 100 கிலோ சால் சோரியா லவர்ஸ். The salt seed is a very important component for chocolate. அது இல்லைன்னா சால் வந்து எங்கேயுமே இருக்கு, ஒன்லி மார்ஸ். சால் சீட் வந்து கலெக்ட் பண்றது நம்ம ரைட்டிங் அவங்க சேவ் பண்ணிருக்கோம். ஒரு 100 கிலோ கலெக்ட் பண்ணிடுவாங்க. கொண்டு வந்து அந்த அவுட் சைட்ல பாஸ் கொடுத்தாங்கன்னா 100 ரூபாய் கிடைக்குது. 100 ரூபாய் அந்த அந்த மிடில்மேன் கொடுப்பாங்க. He is happy with the 100 rupees. அவருக்கு வந்து அங்க மாவளம் அங்க கேட்டதும் கிடையாது. He is very happy with whatever. அந்த கம்பெனி இந்த மிடில்மேன் அப்படிங்கிறது சோ இந்த சட்டம் என்ன சொல்றது, அதே மாதிரி நம்ம கன்வென்ஷனல் பேலன்சிங் கெமிஸ்ட்ரி, நீ வந்து உன்னோட நீ கலெக்ட் பண்றது கரெக்ட், பட் ஓவரா எக்ஸ்ட்ராக்ட் பண்ணிட்டீங்கன்னா அந்த ஸ்பீசிஸ் அழிஞ்சு. அதை தடுக்கிறதுக்கு என்ன பண்ணணும் அப்படின்னா, நீ வந்து உன்னோட லாபத்துல ஒரு 0.5% கம்யூனிட்டிஸ் அவங்களுக்கு ஒரு போனஸ் மாதிரி ஒரு இன்சென்டிவ். அவனுக்கு வந்து சப்போஸ் அவனுக்கு 100 ரூபாய் கிடைக்கிறது, நீங்க ஒரு 10 ரூபாய் கொடுத்தீங்க, உங்க லாபத்துல அவனுக்கு ஒரு 10 ரூபாய் கிடைக்கும் ஒரு நாளைக்கு. இந்த ஸ்பீசிஸ்னால எனக்கு வந்து லாபம் கிடைக்கும். ஓவர் அட்டென்ஷன் என்ன பண்ணுவாங்க, இந்த 100 கிலோக்கு பதில 90 கிலோலே நிப்பாட்டிங்க. அவனுக்கு 100 ரூபாய்க்கு மேல அவனுக்கு ஆப்பர்ச்சுனிட்டி அப்படிங்கும் போது 90 கிலோ நிப்பாட்டிக்கும். மீதி 10 கிலோ அந்த மரத்துல. சோ கார்பரேட்ஸ் வளர்றது தப்பு கிடையாது. It has to grow because it is directly benefiting the community. அவங்க இல்லைன்னு வச்சுங்க, இந்த மக்கள் வாழ முடியாது. 11 கோடி மக்கள் இருக்காரு ஃபாரஸ்ட் நம்பி, ஃபாரஸ்ட் ப்ரொடியூஸ் மாத்திரம் நம்பி 11 கோடி. அவங்களுக்கு ஒரு லைஃப் வேணும், அவங்களுக்கு லைஃப் அவங்களுக்கு பணம் த்ரூ மிடில் மேன். இந்த லாபமும் வந்தது அப்படின்னா இட்ஸ் கால்ட் பெனிஃபிட் ஷேர். தே ஆர் ஷேரிங் தட் பெனிஃபிட். அந்த இந்த ஏரியால எக்கனாமிக் டெவலப்மெண்ட். இந்த சட்டம் வந்து இட்ஸ் a classic link between corporate India and rural India. Where corporate India can help rural India through families.
Sir. Sir.
I think that the environment is not the part time. The sector is not good. I think. We don't need to provide and providing food and direct food right. It is possible to that in the.
ஒரு ஸ்பீசிஸ் பாத்தீங்கன்னா ஒரு டைப் ஆஃப் லயனை கொண்டு வந்து வெஸ்டர்ன் காட்ஸ்ல விட்டீங்கன்னா அதனால வரும், டைகரை கொண்டு போய் கீரில விட்டீங்கன்னா அதனால வரும். லயன் lives in vast open land. அதுக்கு போய் ஷெல்டர் கொடுத்தீங்கன்னா அதை ஓடி போயிடும். It likes to thrive in vast open land. ஏன்னா lion only in Africa. Major population of lions is found in only Africa. So it requires open.
பிளானிங் பண்ணுவாங்க. மினிமம் பிளானிங் இருக்கு. இட்ஸ் நேஷனல் பார்க் அப்படின்றதுனால ஒரு மினிமம்
அதுதான் நான் ரொம்ப நேரம் பார்த்துட்டு இருக்கேன்.
கேள்வி கேட்க போறியா? சரி ப்ளீஸ்.
Good afternoon, sir.
Sir.
Sir, I identified one particular area. That particular area, a particular group of birds flying morning and evening daily. In that particular area, some companies are raising to build the company. The companies are emitting toxic gases in the environment. The gases are affect the air spark. It is possible to prohibit the company to raise in that particular area.
வரமாட்டேங்குது சார்.
இந்த क्वेश्चन அப்புறம் முடிச்சலாம். கம்பெனி பேரை சொல்ல வேண்டாம், எந்த ஏரியான்னு சொல்ல வேண்டாம். பட் இப்படி ஒரு சிச்சுவேஷன்ல when there is a visible pollution of air or water on land, I think environmental protection is essential much. So if you find you want from your side to help the birds, the bird population, you can always report the matter to the environment department. Pollution control board, state pollution control board. Over the administrative department for this environment protection. அவங்க கேஸ் எடுக்கலாம். அவங்களோட பர்மிஷன் இல்லாம எந்த இண்டஸ்ட்ரி ஃபங்க்ஷன் பண்ண முடியாது. If you think that you want to give a complaint, that's two departments, the forest department. One is the environment department, pollution control. Ensure that someone will take note of or take cognizance of your letter. And what the cognizance எடுத்துட்டாங்க. Either it will affect the wildlife protection and also the economic protection. And what the cognizance எடுத்தாங்க. So the company will have to take a recourse. அவங்க வந்து அதுக்காக company process இருக்கு. No. See, ஒரு பார்க் வந்து எந்த ஏரியா சொல்லுங்க. இல்ல அப்படின்னா இவங்க give a recommendation to the forest department. பட் என்ன ப்ராப்ளம்னா ஒரு ஏரியா வந்து வைல்ட் லைஃப் சேஞ்சுரியா நேஷனல் பார்க் டிக்ளேர் பண்ணாங்கன்னா அங்க உள்ள ஃபெசிலிட்டிஸ் எல்லாம் மாத்த வேண்டியது ஏரியா. அது வந்து மக்கள் வந்து பயங்கரமா எதிர்பார்ப்பாங்க. அப்போ வந்து you have to just report to the concerned department, environment department, pollution control board or forest department. அவங்க they will issue notice to the company. அவங்க அந்த இதெல்லாம் மாத்தணும். That is the only way. National park or sanctuary ஆடுனாங்கன்னா ஸ்டேட் கவர்மெண்ட் ஒன்ஸ் நோட்டிஃபை பண்ணிட்டாங்கன்னா ஸ்டேட் கவர்மெண்ட் பவர் கிடையாது. அதுக்கப்புறம் டீ நோட்டிஃபை அதான் அதுல இருக்கு. State government has power to notify an area as a wildlife sanctuary or national park. Once declare பண்ணாங்கன்னா no going back. அவங்களுக்கு திருப்பி டீ நோட்டிஃபை பண்ணணும். கேட்டுக்கிறியா? Just to be done only. Sir, like பண்ணுங்க. அதெல்லாம் personal so நீங்க you take the lead and he was already நீ கேக்கும்போது ஃபைனல் அந்த உயர்வுனா நல்லா பண்ணிருக்கேன் ஒரு so அத நீங்க சேர்ந்து You are it is only one life. 100 years. 100 years. So it is one life. So a portion of our life we can always contribute for our society.
Sir. Sir, I have one final question regarding this. Sir, Clean Ganga project. I heard that it has improved gangetic dolphins population and has done better to gangetic dolphin populations. Does it have improved many lesser known species like gharial, gangetic shark, which is a very rare species, and many other unknown species like gangetic soft turtles? Does it has improved the conditions of other lesser known species?
Gangetic dolphin. Super. Gangetic dolphin.
Yes. National aquatic.
Very fantastic. Actually, it is the apex species of freshwater ecosystem. Freshwater ecosystem effectively number tiger is the apex of the coastal ecosystem. So, the apex species is good. That means the entire species below it is also thriving. So, if gangetic dolphin is thriving well under the Namami Ganga project, that means feed sufficient feed. So that means if the apex species of a particular ecosystem thrives well, all the entire and the ecosystem species they are very very happy. So one species apex species thrive other species India for tiger to thrive in a coastal ecosystem, square kilometers, but man population population gangetic dolphins thrive needless. So that means that other species thriving well. Yellowstone Park
Introduction.
people, they are very kind-hearted people. I was not in the the wolves in digest நம்ம முன்னாடி சாப்பிடுது இந்த பண்ணி விட்டாங்க அவங்களுக்கு தெரியல எவ்ரி ஸ்பீசிஸ் இந்த ஃபுட் ஃபார் அனதர் ஸ்பீசிஸ் தெரியல. இது என்ன ஆயிடுச்சு, எல்லா உல்ஃப்ஸும் அந்த ஏரியால இருந்து கொண்டுட்டாங்க. அப்போ உடனே இந்த ஹெர்பிவர் பாப்புலேஷன் இன்க்ரீஸ் ஆயிடுச்சு. ஹெர்பிவர் பாப்புலேஷன் இன்க்ரீஸ் ஆனோட அது என்ன பண்ணிடுச்சு, அங்க உள்ள ஒரு அட்டகாசமான ஃபாரஸ்ட் வளர விடாம திரும்ப உள்ள ஒரு சீட்லையும் கூட வளர விடாது. கம்ப்ளீட்டா இதாகி ஒரு நல்ல ட்ரைவிங் ஃபாரஸ்ட் பிகேம் பக்கா கிராஸ்லேண்ட் அன்ப்ரொடக்டிவ் ஈகோசிஸ்டம் ஆயிடுச்சு. அதை ரிவர்ஸ் பண்றதுக்கு இட் டுக் லாட் ஆஃப் இயர்ஸ். தட் இஸ் த Yellowstone நேஷனல் பார்க். அது ஒரு ஸ்டோரி ஆஃப் ஹவ் வி ஷுட் மேனேஜ் அ ஃபாரஸ்ட். நம்ம ஓவரா ஹியூமன் இன்டர்வென்ஷனால ஒரு பர்டிகுலர் ஸ்பீசிஸ் நம்ம எலிமினேட் பண்ணணும்னு நினைச்சீங்கன்னா வில் கிரியேட் அனதர் ப்ராப்ளம். ஓகேங்களா, ஒரு ஃபாரஸ்ட் டிஸ்ட்ராய் ஆயிடுச்சுன்னா என்ன ஆகும், அந்த ஏரியால தண்ணி இருக்காது. ஃபாரஸ்ட் இஸ் த ஸ்டோர் ஹவுஸ் ஆஃப் வாட்டர். அந்த ஏரியால உள்ள மக்களுக்கு ஃப்ரெஷ் வாட்டர் கிடைக்காது. ஃபாரஸ்ட் இஸ் த சயின்ஸ் ஃபார் த
சார்
If you have any doubts, you can ask personally to the sir.
Dear participants, The lunch will be provided for you at the upstairs of this hall. And after the lunch, all should assemble in the hall for the next session at 1:50 p.m. And one major thing, the attendance for our students will be provided at the end of the event, that is evening, with their respective tutors. Thank you.
You have to ask me assembly by 2 p.m. That one person is
ஏதாவது பண்ணணும்னா அவருக்கு
Hello, hello there. Hello. Hello, hello there. Can you hear me? Hello. Yeah, I can hear, but it is echoing. It's not very clear.
Sir. Volume on.
Now I can clear better. Thanks.
ஒண்ணுமே பண்ணலங்க.
Can you see my screen?
Sir.
Hello.
Sir. Sir.
Hello. Yeah, I'm ready to share. Do you think, can you see my screen? Presentation screen?
இப்பதான் வரல.
How about now? Can you?
Sir, now we are visible to your screen, sir.
Okay, thank you. So I'm ready to present whenever you are ready. You people are ready. Thank you. Hello, yes, sir. How are you? Sir. Sir. Thank you, sir. Thanks for the invitation.
இங்க பேசுங்க.
பக்கத்துல வந்துக்கோங்க சார். இது இப்ப யாராவது கொஸ்டின் பண்ணாங்கன்னா கேக்குதான்னு தெரியல. நீங்க தூரத்துல இருந்து கேக்குதா? சப்போஸ் நல்லா வரலன்னா நான் வீடியோ கட் பண்ணா கொஞ்சம் பெட்டரா இருக்கும்.
நம்ம கிளாஸ் பசங்க எல்லாரும்
is from now general now. Then there is around Okay.
Sir, if anyone is asking question, I think they have to come close to the laptop. Otherwise, I cannot hear.
Okay, sir, okay.
அதான் சார்
சார் யாராவது கொஸ்டின் கேட்குறாங்கன்னா கம்ப்யூட்டர் பக்கத்துல வந்தாதான் கேட்கும் போல இருக்கு. நீங்க தூரத்துல இருந்து கேட்க மாட்டேங்குது.
சார்
starts now. Adopting on the regulation of saying.
Good afternoon, one and all gathered here. Now, we are in our today's second technical session. Our today's chief guest of this session is Dr. N. Balachandra. He is a scientist, field therapist, California, USA. His topic is regulation of gene expression by nuclear receptors and transcription factors.
Am I good to go?
Okay, now we can start. We will discuss that. You can start your lecture now.
Okay, thank you. I think if you're talking close to the laptop, I can hear better. Thank you. Yeah, thanks for the invitation. It's my honor to present some of my key work here. I was in Jamal until 98 to 2000. I was the first student of SSR, Sheikh Umar Shahid sir. So there first time I tried to get familiar with molecular biology. Then I got interest in molecular biology from then onwards. Then I got my PhD in molecular biology in South Korea. Then I came to US and I did a postdoc, a lot of things for almost 12 years. Now I'm working in industry as a scientist. So today the title of my topic is regulation of gene expression by nuclear receptors and transcription factor. Before going into detail, I will give you a brief introduction about what is signal transduction. So if you look at any receptor, either membrane receptor, it is a cell membrane. Just I made a cartoon to better understand. So then this is the nucleus and then cytoplasm. You could see that if any signal, if a receptor is getting, like if a hormone is binding to a receptor, like GPCR in the membrane, then it transmits the signal through the second messenger. Like a lot of second messengers are there, like kinases and other kind of adenyl cyclase, this kind of second messengers, and then the hormone can bind to, or ligand can bind to a nuclear receptor or hormone receptor. Then it gets the signal, it gets activated and it goes to the nucleus. And in the genome, you could see the promoter region. They will have its own responsible binding element called hormone receptor element. In general, I give HRE, so they can bind to this binding site and then they can activate the gene, a responsive gene. So this way, every gene is regulated by nuclear receptor or transcription factor. by binding onto the promoter region. And if you see that when it is get activated, then it expresses the mRNA obviously, and protein is synthesized in cytoplasm, and then the further protein is modified, or final protein it will be ready for the biological activity. This is how each typical gene is controlled or regulated by nuclear receptor or transcription factor. So this is a simplified cartoon to show the signal transduction here. And if you, I will go a little deeper. I don't want to go very much deeper into my area of work. Just I want to give an overview of each projects, and then I will cover a few of the key data with you, all of you people. I won't take much time also for the presentation, but I will be happy to take question at any time, and then I'll be ready to talk in detail. If anyone wants to know anything particular, I will be ready to talk in detail. And this is about the typical structural organization of nuclear receptors. In general, a typical nuclear receptor, hormone receptor has nuclear or hormone receptor are same. So they can they have a AF1 domain and then AF2 domain at the C terminal. So you could see that DNA binding domain, they will have that, and then ligand binding domain. This DNA binding domain or ligand binding domains are the key, and these two binding domains can be linked with the hinge domain. This is a typical structure of a nuclear receptor. But whereas I was working on, our lab was up a nuclear receptor lab, and we were studying its role in various biological functions and during my PhD. So typical orphan nuclear receptors are like this. Although they have a ligand-like binding domain, but they don't have a typical DNA binding domain. That's why they are called orphan nuclear receptor. And then we were mainly working on SHIP, SHP, small heterodimer partner, and then DAX1. These two, all these nuclear receptors can be nomenclatured by NR0B1 or NR0B2 like that. And my study, I focused on this orphan nuclear receptor called DAX1. So I split my talk into 3 different parts. Each part I will cover very few data, very few key data to focus on each topic. I don't want to take a lot of data. I don't want to show so many data in detail, but I will show some key data to cover each topic. First topic I will cover role of nuclear receptor DAX1 on HNF4 alpha. This is also another hormonal receptor, nuclear receptor during the nuclear gluconeogenesis. And then second part, I will cover the identification of GLIS2 interacting protein. And then the final part, I will cover functional characterization of a gene called JASF1. The first part, I will be focusing on the DAX1, role of DAX1 on HNF4 alpha during gluconeogenesis. So this is one of the key data I showed. This is published in JBC long time back when I was in our PhD time, around my time in Korea. So this is a typical report region assay. We call it as a report region assay. One promoter region is, hello? Everything is good? Hello?
Yes, sir.
Sir, is it everything is good or? Hello? I can't hear anything. Sir, are you trying to talk something? Is it, sir? Okay, then I'm good. Thank you. I thought you were trying to say something or ask any question. So if anybody want to ask a question, you can raise your hand from there, and then I can. So this is the report regime assay. So here I cloned the H104 alpha binding site in report regime vector. So report regime vector called luciferase vector. So luciferase assay here I'm showing. And so this is a relative light unit here. You could see when I co-transfect this promoter along with the H104 alpha, you could see this is the basal, nothing is expressed in the cells. So when I express H104 alpha alone, you could see it is activating very high. So this indicates that H104 alpha can bind to this promoter region, this multi-copy binding site H104 alpha region, and then it can drive the luciferase gene expression. So this reports the gene expression. That's why we call it as a reporter gene assay. And when it is co-activated, if I co-express DAX1 along with H104 alpha, you could see the H104 alpha activity was drastically suppressed by DAX1 expression. You could see here dose dependently. And then this is I used a SHIP as a positive control because this one also, it was around that time it was previously published. So I used this one as a positive control. So this is a typical reporter gene assay where we can get to know H104 alpha transcription activity here, and then when I co-express, it is suppressing. So then next I wanted to know whether this changes in activity is due to the changes in changes in protein level. So I checked the protein level of H104 alpha, and then DAX1 here, you could see the proteins level was not changing. H104 alpha protein level almost same for all the condition wherever I express. Plus indicates the expressed wells. So when I express with the DAX1, it doesn't change the protein level. It's not due to change in protein level. So it is change in activity, promoter activity. So this result shows that H104 alpha transcription activity was decreased by the DAX1 expression, co-expression. And then next I wanted to know whether it is due to the direct interaction. So here you could see the co-immunoprecipitation assay data here, this one. So for co-immunoprecipitation assay data, it will show whether the two proteins are associated. So here you could see when I Western IP with the DAX1, one antibody, and then Western blot with another antibody, H104 alpha. So it shows very clear interaction here, association here. So this indicates that these two proteins were associated in vivo in the cell. So, and then another way of showing this interaction, we can show the co-localization. Here you could see the GFP, this one GFP DAX1, and then ICEH-2023 alpha. So GFP DAX1 obviously green fluorescent protein, green color, and then ICEH-2023 alpha. I did a conjugate secondary antibodies conjugated to give red color. So you could see this red color indicates H104 alpha. When I overlay these two images, wherever you could see the yellow color, it means these two are co-localized. So you could see mainly they are co-localized in the nucleus. So this indicates that these two proteins are co-localized in the nucleus mainly. And you could see then next I wanted to know whether this interaction is mediated through any cofactors. So here I used PGC1 alpha. This is PPAR gamma C alpha, it is called, this protein. This is a co-activator. When for H104 alpha, it is previously known. When we co-express when we express HNF4 alpha, as you can see from this previous figure, it was activating. Same way here also it is activating when I express the HNF4 alpha with the promoter. And then if I co-express HNF4 alpha along with the PGC1 alpha, it is further activating. So this indicates that this is a co-activator, and it is well-known co-activator of HNF4 alpha. And this HNF4 alpha co-activation is very high here. And then when I co-express DAX1 in a dose-dependent manner, you could see it is dose-dependently decreasing. So this assay indicates there is a co-activator and DAX1 competition. So it means DAX1 is acting as a co-repressor here and for competing with the HNF4 alpha binding. That's why we are seeing this effect. And then this further, we can confirm this with another kind of experiment here. You could see this is this experiment called GST pull-down assay. So where I purified the GST HNF4 alpha protein, and then I incubate H8 tagged PGC1 alpha. This is cold, and then I have a hot one. This is S35 labeled. This is isotope. If we are in vitro translating with this isotope, this will label our protein when we are in vitro translating. This is like whenever it is adding amino acid, whenever it is serine coming, then S35 will bind there. So that's why we see the signal. This is a image radio isotope image. So you could see that this is here. And then when I did cold one, unlabeled in vitro translated one in a dose-dependent manner, you could see this is dose-dependently, this signal is going away. So this further support this effects, like these two protein are like PGC1 alpha and then DAX1. They are competing with each other for binding to HNF4 alpha. And then whenever this one, we found that there is a repressive effect of DAX1 interaction, and then mechanism, how it is working. So this all together, I studied the molecular mechanism here, how a gene is regulated. And then I wanted to show the physiological relevance of the interaction or this co-activator or DAX1 competition. So for that, I wanted to focus on the glucose metabolism, because HNF4 alpha is known to play a role in glucose metabolism. So that's why I try to give you a small intro about this glucose homeostasis. So you could see when during short-term fasting, glycolysis gets activated. Prolonged fasting, gluconeogenesis gets activated. And when it is feeding time, there will be a glycogenesis, and then lipogenesis gets activated. So here we consume the sugar in our body. Here we will get depleted when it is when we are fasting too long. Our glucose is from our body gets depleted, so it needs to find a source. So it goes for a gluconeogenesis process called glucose synthesized newly from other source. So, and when we are in feeding state, regular feeding state, glycogen genesis is activated. Glycogen is stored in the liver and other parts, and also lipogenesis also gets activated. Lipid metabolism gets activated, and it is stored in the liver and other parts of the body. So prolonged fasting time, gluconeogenesis gets activated, like overnight fasting if we do, or more than 10 hours of fasting, they gets activated. These two are the key enzymes and we can focus on these two reports. Okay, there is a gluconeogenesis activate gluconeogenesis. So this two enzyme called PepsiK and G6-based glucose 6-phosphatase and phosphoenol pyruvate kinase carboxykinase. These two are the key enzymes for gluconeogenesis, and these two enzymes are regulated by multiple nuclear receptor and transcription factor. You could see here. You could also see the HNF family, HNF alpha, HNF3 also there. And the final figure of this first topic, I will show the physiological relevance here. This is in the primary hepatocyte. So when we treated the mice with postcholine or insulin for a while, and then we sacrifice the mice and get the primary hepatocytes out of the mice. So you could see here the PepsiK gene expression in primary hepatocyte after the treatment of postcholine. Postcholine is a compound that can activate the cyclic AMP pathway. So when cyclic AMP pathway is activated, gluconeogenesis is very much high. So when we fast for overnight, so gluconeogenesis is high because the PepsiK and G6-base enzymes are high. So here they are high when body start secreting the insulin. When we get the insulin, when we start feeding the insulin, our body will secrete particularly in the body, pancreatic beta cells, they secrete the insulin, and then they can suppress the postcholine mediated activity. So here you could see PepsiK and G6-base activity is completely gone when insulin is treated. And this one, I made the adenovirus around that time, like 15 years ago, GFP adenovirus and then adenovirus DAX1. You could see the overexpression of DAX1 in this condition. This is a control condition. If you compare this one with this one, very significantly decreased here and here. So this indicates that overexpression of DAX1 significantly decreasing the postcholine mediated gluconeogenic gene expression. And this is a control to show my virus is working. So it is very highly expressed here, nothing. When I express only the vector alone. And then here DAX1, you could see a very huge expression is there. And here the glucose output in the cells, you could see postcholine, the glucose output is higher, known to be higher when we have a cyclic AMP signaling or gluconeogenesis is happening. Our cell send the glucose out of the cell. That's why we check the glucose output. And this output also decreased when I overexpress with the adenovirus DAX1. And this is mRNA level, this one glucose output by ELISA. And here northern blotting to show the same thing here. PepsiK gene expression, postcholine, PepsiK is high, and then insulin completely gone. Here the postcholine and then adenovirus infected cells, you can see very significantly decreased, and then alone only compared to control, and then postcholine effect also decreased. So this indicate that without the insulin also adenovirus DAX1 overexpression was efficiently decreasing the PepsiK gene expression. And next, I was checking the blood glucose level. It is in vivo in the mice. After infecting with the DAX1, you could adenovirus. You could see this SHP adenovirus. Previously, we made this adenovirus vector. So this is the control I used. Here, DAX1 also in vivo in the mouse. You can see it is also decreasing from here. You could see that DAX1 overexpression is decreasing the glucose, blood glucose level in the fasting condition. And here, high-fat diet induced mice I used. Then you could see it is known to have a high level of PGC1 alpha expression and this key enzyme expression, and they were also decreased by DAX1. So overall, this my study here, I will show in a cartoon, like insulin signaling and feeding condition, but I didn't show this data here. I don't want to show a lot of data. Only key data I'm showing here. DAX1 is induced, and then when it is induced and when it is going inside the nucleus, it competes with the PGC1 alpha, and then it decreases the gluconeogenic gene expression. So this is one of major key finding I did in during my PhD. And this is summary of the first topic. And then second topic I will go, this is about another transcription factor. It is not the nuclear receptor. They also involved in the gene regulation, but they are not in the nuclear receptor family. So one of the transcription factor called GLI similar protein 2, that is also called GLIS2. So I try to identify GLIS2 interacting proteins in this project. So I will give you a little intro about the GLIS family. You could see this is the GLIS family, are Kruppel-like zinc finger proteins family, this one. And you could see GLIS3, GLIS1, and GLIS2 are here. And this is a phylogenetic tree. This will show how they are interrelated with each other. So you can see GLI family are together and GLIS families are here together. And when we knock out this GLIS2, they are known to cause apoptosis and epithelial mesenchymal transition, and then T and B cell B cell infiltration, as well as macrophage infiltration. So all together they can cause this kind of a phenomenon, and then they cause, I don't want to go deeper into this mechanism, but this loss of GLIS2, when we knock out the GLIS2, it happens, they will make the kidney to become a fibrotic kidney. They become very shrunken, and then we can see by naked eye also, we can see it is very, very much different. So this is called a nephronoptosis or renal failure. So when we are knocking out only this gene, it causes the biggest renal failure. So this one, our group reported about this. And then this is my staining data. You could see when wild type mice, very nice and clean kidney staining. This is H&E just for morphology. I didn't stain with anything else, just to show the morphology. When we knock out the GLIS2, it is getting worse from almost 2 months. I can see that here also I made this data little and getting worse. And when it is coming close to 1 year old mice, very bad shaped kidney. We can see literally from by looking at the kidneys, comparing control and the knockout, we can easily see there is a clear damage or renal failure there. And when I'm always interested in focusing on the molecular biology. So when I joined in NIH there, so I wanted to look out the novel interacting protein of GLIS2, as well as modification, like post-translational modification of GLIS2. This was my aim, and then I wanted to focus on this work. So for this, I've created few cloning. I made that GLIS2 gene. First, we can take out any kind of gene. If we have a mRNA from there, we can get from there. We can clone any gene. So I got this sequence out from a cell line, and this is mouse GLIS2 gene. And then I cloned in a CME flag vector. So those days I clone with enzymes, 2 different enzymes in one vector. I can put one enzyme in one end, another enzyme in another end. And when I digest the vector, I can put this gene here. So I did that. This is a very classical way of cloning. Nowadays, more recent advanced cloning is there. And I don't want to go into deeper there. And then when then I moved this to another vector called CMA-HA tag vector. There it has it will get a HA tag also. These two are tags, flag and HA. And then I moved this one again to another vector like called PIRIS to GFP vector. So what this vector has is like, apart from these 2 tags, it will have a IRES sequence, internal ribosome entry site sequence. And and then along with that, it will have a GFP in the vector. That's why PIRIS vector. So this vector will express flag, and this transcript will have flag, GLIS2 gene, and then HA, and IRES sequence, and then GFP here will be there. But when getting translated, this IRS sequence will allow the ribosome to bind here, this internal ribosome entry site. Then it will get only this site will be expressed for GFP, and then this one will be separate. So this way, this is like a selection marker for this vector. I cloned this. Is a complicated cloning around that time. So I did this cloning here, like it will be like cloning that, and then I transfected into a mammalian cell line called ICEH-2023 cells. And they are freestyle cell. We can use as a monolayer also in the plate, and then we can use in a suspension like shaking media also. So and then stabilize them. This is a way of making a stable cell line. So when we have this stabilized with geneticin, because one of the vector has the final vector will have this marker will be expressed. So whatever cells they have this expression vector is there, so they are resistant to this geneticin. Rest of the cells will be killed. And then this is how we can select a typical. This is one of the way to select a make a stable cell line. I made a stable cell line, and then I did flow cytometry analysis to pick top 10 positive GFP positive cells, and then I went for large scale culture, like 1 liter of culture, where we can get 1 billion of cells. So it's a huge amount of cells because I need for immunoprecipitation. So I did immunoprecipitation IP with the flag conjugated beads, and then next I did this beads after IP. Then if I incubate with the flag peptides, this will release the flag conjugated with the other proteins, and then that will be in all the protein complexes will be released in the and it won't be attached with the beads, and then I can get rid of the beads, and then further again IP with the HA. That's why it is called tandem. One side flag, and then another side HA. One IP, second IP I did, and then stain the gel, submit the gel for mass spec analysis. In the mass spec core, I submitted where they cut the gel into each lane into pieces, like 12 to 15 pieces of gel, and then they do the mass spec analysis. Whatever protein are there, we can identify from there. So these are all, I just give you a few here. I got like 200 proteins, but I chose a few here. These are all the protein I got from mass spec analysis. And then I don't want to say go into deep detail for each protein. And this is a known complex. Previously, we know they have published paper, they show the complex. And I also got the known complex along with the other new novel protein. So this is, I hypothesized the complex based on this data. Here you could see that there could be a possible complex between GLIS2 and interacting proteins here. And then to further confirm this interaction, I had to go do other kind of experiment like co-immunoprecipitation. I did co-immunoprecipitation. I just tell in briefly here, this co-IP shows CTBP2 and CTBP1 are interacting with GLIS2. This one, I used the GLIS. Here you could see here, wherever I co-express GLIS2, and you can see the signal here. So, and then here for KDM1, all of them are, we need to do a lot of multiple cloning here. So I did the multiple clonings for each vector. And this is another KDM1 also interacting with another protein in the complex I showed in the previous slide. They are also interacting here. And here also you could see the co-rest, another protein also interacting. This is just co-IP data. And here I show the co-IP with all other family, the CTB GLIS2 family, and then with CTBP2. Only GLIS2 is interacting, not GLIS1 or GLIS2 is interacting. And here I show the co-localization of GLIS2, this one, and the CTBP2 here. Actually, CTBP2 is MIG-tag here, mislabeled here. So if we are any labeled protein, any tagged protein, secondary antibody we can choose for different colors. So here I choose for green, and then here I choose for red. The overlay indicates here, yellow indicates the co-localization of these two proteins. And next, I want to show the functional meaning of this interaction. You could see there's CTBP2 overexpressed and further activating the GLIS2 activity, transcriptional activity. I used a insulin promoter, insulin gene with the reporter gene, and GLIS binding site with the reporter gene. You could see both are activated, and when I co-express CTBP2, further activated. So this data shows like GLIS2 acting as a co-activator here. And then here, in this data, I compared with the other family. Only GLIS2 was co-activated by CTBP2, not GLIS1 or GLIS3. So this is a cartoon shows what I did. And then I wanted to show whether it has any interaction with the other protein like cofactor like PCAF or P300. So I did, to show that I had to do interaction assay in vivo IP. So this indicates here PCAF is interacting in two-way. This is two-way IP. And this one CBP alpha also interacting with GLIS2. So in summary, they have a complex CTBP2 and PCAF because they are co-activating. That's why they are bringing co-activators like PCAF or CBP. They are co-activators. So that's why GLIS2 activity is going up. I showed this, proved this with the previous experiment. So this is all about that second topic summary. I didn't show much of the other data to take more time. It will take only one topic. I can present a lot of data, but I want to show the key data for general audience. And this is the third and final topic, where I created a knockout mice. This is the gene for this one, JASF1, and this gene called JASF1, tip interacting protein, TAC1 interacting protein. So they have this zinc finger domain and then this TAC1 interacting domain. So these are all the previous literature shows their association with the other factors and their role in their physiological role. And this is the cartoon shows this one previously we found this one as the interacting partner of TAC1, another nuclear receptor, and then they have a similar expression pattern, and they have these two, like TAC1 and JASF1 has similar expression pattern, and this JASF1 SNPs, like single nucleotide polymorphisms, are associated with diabetes. And another role also, they are highly expressed in beta cell, and so altogether this indicates that this JASF1 may have a role in obesity, diabetes, liver steatosis. So that's my the aim I wanted to check here. So after creating the knockout mice, but the knockout mice took like 3.5 years to get the first knockout. So it was very complicated knockout. So this is the 5' region of the gene, 3' region of the gene. We have a reporter cassette with the selection marker, but I don't want to explain everything in detail. If you look at FRT, green. So this is a site. If we are breeding the mice with the flip mice, so they can cleave this site and remove this region. And if I breed with the cream mice known to express the recombinase known to cleave the LOXP site. So that is how typical knockout were created 10 years ago, but now CRISPR is excellent, so we can do faster and better and easier technology. That also I have done. I'm not showing here, but this one, the classic way of making a knockout. We can put a reporter cassette, but we were having trouble in getting a knockout. So that's why we came up with this strategy. First, breed with this first heterozygous mice with the like TX mice, targeted mice with the flip mice. So when we breed the mouse having this one with the other mice having the prit side, this will be removed. That's what. And then next we get FX, we label like FX mice, and then when we are breeding with the Cree, these two will be removed. Mainly these two will be removed. Wherever LOXP is there, it will be removed. Already this LOXP is gone, so this side is removed. This way, you could see this is the targeting region gets deleted, and the protein, the expression will be out of the frame. So this JASF1 protein won't express. mRNA won't be there, protein won't be there. This is how we get the knockout. Where I got the knockout mice. First knockout mice took 3.5 years to get first knockout mice. Then I compared the staining here with just before breeding to LOXP with LAGZ gene. So this XGAL staining shows, blue color shows like JASF1 is expressed. And here it's at the mRNA level. I compared the knockout mice. Here you could see the wild type and knockout very almost gone. There's the Jasafan expression, but just Jasafan is known to express highly in testes. So here you could see very drastic decrease are completely gone. But I just here the values I wanted to show this base level also. That's why I zoomed in very much, but it is completely gone. Here northern blotting, those days I used to do a lot of northern blottings to show the gene expression at mRNA level with a labeled isotope P32 here, not like protein I labeled with S35. I showed in GST pull down assay. So altogether, this data shows that my gene is knocked out. And then this is a cartoon I want to show. If we are taking more high-fat diet, people like prolonged high-fat diet feeding, people or mouse or anything, any animal, other animals also get obesity and insulin resistant eventually, most of them. That's why hyper diet induced obesity and insulin resistant I want to focus on. So around this condition, our liver gets fatty, our adipose get inflamed, like immunological action, immunological responses happens, and then beta cell eventually slowly beta cell function gets deteriorated, gets bad, and then there will be a beta cell failure or beta cell loss also. That's why initially people when they are getting diabetes, and they can just metformin tablets they can get, and they can try to decrease the blood glucose level, but eventually if they are keep on eating the hyper diet, there will be loss of beta cell. When beta cells are lost, completely lost, then insulin won't be there, so they have to take insulin. Or some people, and the natural insulin won't work, that's why artificial or lab made insulin will work for them. So this is a typical like diet induced obesity model. So if we are feeding the mice with hyper diet for 12 weeks, they get obese compared to normal diet, and that's a hyper diet model. And then I want to put both now wild type mice and then my knockout mice for in hyper diet, and they both gain weight, but knockout in the red you could see very much decreased the weight. They were not gaining weight like the wild type mice. So this indicates that Jasafan knockout is protecting, or it is resistant to diet induced obesity. And that one we can also check in vivo in different way, insulin tolerant test or glucose tolerant test. So it's like if people are getting diabetes fasting condition in the early in the morning, they go to doctor, they check the blood glucose level, same way I did in the mice also. People used to do in the mice also. They can starve the mice for overnight, and then if we are giving glucose, it will go high. This glucose level will be high because insulin resistance will be there. And when compared to wild type, knockout mice has less glucose tolerance. So overall, this my last part also covers the Jasafan mice was created, and when I put in hyper diet induced obesity model, it plays it shows that it has a vital role. It is significantly decreasing the weight gain as well as glucose tolerance. That is the report. That's the one another story I was presenting. that's all about my talk. I think I'm on time. If anybody has any question, please come close to the a laptop and then ask me any question. Thank you. Any question? Hello? I didn't hear anything.
Sir, good afternoon.
Yeah. No need to be just you can call me Bala. Just you can ask me question.
Okay, I have some. Yeah. I have some questions regarding that knockout method. So
Nice.
knockout method.
Okay.
So you used this LOXP three LOX mechanism, isn't it?
Yeah, 3 lakhs pieces are there.
Okay, I heard that this three LOX mechanism is for deleting tissue specific gene expression, isn't it?
I'm sorry.
Uh,
Okay, could you
whether this method is used to knock out the gene in every cells or whether it is tissue specific.
this is not tissue specific. If it is tissue specific, I have to put a unique promoter for tissue specific. Here, it's not tissue specific. It is in every PGK gene is driving, so it is known to be expressed in every tissues. It's a global knockout. It's not tissue specific.
Okay.
Good question. Good question. I'm glad you are asking detail about this one, whether it is tissue specific or global. Very good question. It is a global knockout.
Okay, in case of other methods, apart from three LOX, what are other methods for knockout? That's the method name. I don't want again. What are other methods?
You mean the knockout?
For knockout.
In knockout, we can do multiple other ways. We can nowadays CRISPR, TALEN, zinc finger based knockouts are there. So we can do, if I use if I design a guide RNA one side here, that also I didn't show that data. That's a big another project in stem cell biology. So if I design one guide RNA here, another guide RNA here, or if you look at only this portion instead of LOXP side, I can design one guide RNA here and then another one here. This way, these two guide RNAs will cleave this site and then remove them. So this way also we can create a knockout with the CRISPR or another technology.
Okay, this last one, that's one gene, because a gene can have multiple forms. So if it has many forms called alleles, then whether we target all the alleles or how can you do that one? Because
which one? Could you repeat again?
A gene can have various forms. For example, class one, have one gene can also have its own forms. So this particular knockout will delete every forms of that gene.
Yeah, a lot of isoforms are there. So even though multiple isoforms are there, if there is homology, here 5' and 3' arm homology is matching wherever it is matching, whatever isoforms they have this homology, then they get deleted.
Okay, then okay, another one thing you said about orphan receptors. So how can I find a ligand for orphan receptor?
Which one? Orphan nuclear receptor?
Orphan receptor you said about
yeah.
How can
I'll come to that. Yeah, that's also good. That's also good question. I will come there. Yeah, here conventional receptor. they have a known ligand or hormones here, like ER, estrogen receptor, like AR, androgen receptor, GR, glucocorticoid receptor. So each receptor has its own ligand. Here, they are adapted orphan receptor. They were orphan nuclear receptor because there were when people were initially investigating each receptor, they categorized them as the orphan nuclear receptor as they didn't have the ligand around that time. But eventually, later on, people started finding the ligands for this receptor. That's why they were adapted from here. These are all orphan nuclear receptors. They don't have the ligands reported, but some of them, people will be working, they would find the ligands for these receptors eventually. Also, there may be a ligands for these receptors, or they may not be a ligands, but whatever it is, this DAX1 and SHP, they won't, even if they have nothing will work because there is no ligand, there is no DNA binding domain, but they have a ligand like binding domain. That's all. Did I answer? That's a good question. I think I told about this, why it is categorized based on the ligand.
That means I want to know the method. What's the technique that they use to identify this?
For ligand?
Ligand receptor.
Yeah, that's a big like one big group of people. They can work on this kind of a project for ligand screening. People nowadays with the duck screening methodology, a lot of different methodology they use to screen multiple ligands in high throughput, and then they can find, okay, if I express if I want to find ligand for this NER77, I can express the NER77 binding site in reporter gene vector in multiple well, and each well I can treat with a different ligand. And then after treating with ligand, I'm telling you in a simple way, and if we are treating with a ligand in each well, different ligand, and then at the end we can wash the cells and do the reporter assay. It will report if it is going high, means that ligand is activating the NER77 reporter gene. So this way we can screen them. So this will be a big project. If I want to spend, I have to spend couple of years to find a ligand for only one nuclear receptor. So it's a big project. So that's why everywhere people are working in different projects, and definitely some groups are trying to find natural ligand or synthetic ligand for each receptor.
Okay, thanks. Final question: what's the difference between qPCR and reporter assay?
What's the difference?
Reporter assay and qPCR.
Yeah, that's very basic. Here, reporter gene assay I'm showing here. So reporter gene assay, it shows the regulation of gene expression, but it is at the promoter level. If you look at a gene will have this 5' side promoter region and regulatory region, and then when all these regulatory elements factors are binding, when it is getting activated, what happens? It will express the mRNA. So here, instead of mRNA expression, we are getting whatever is binding to this side. This is, I put HNF4 alpha binding site only. This is 8x, means 8 copies of HNF4 alpha binding site is here. So if you are expressing HNF4 alpha, so 8 times it can go and bind. 8 HNF4 alpha can go and bind there. And this is a reporter gene. Why we call it as a reporter gene? Because when something is binding to the upstream here of any reporter gene, like luciferase here, so then they get activated, this reports the gene expression. So here it is reporting the gene expression. So we are checking the regulation of gene expression here. But in qPCR, instead of luciferase in the original genome, original promoter will be here. I don't have, if I have the board and pen, I can show that, but I don't want to draw here with my pen here. But this one, if it is mRNA level, when a gene is getting activated in vivo in the genome, here, instead of this one, promoter sequence will be here. And then from here, start site will be there, and then mRNA sequence will be there. So in reporter gene assay, this is just checking the regulation. In qPCR, we will check the gene expression at mRNA level. Now, are you getting that? Good. Did I answer your question? Are you getting that? What's your name? Manikandan, nice questions. Yeah, good. You are trying to learn a lot of things. Very good. This is good. I appreciate the questions. Any more questions?
Yeah. Sir.
Sir? No, I'm not hearing clearly.
Hello.
Yes, sir.
Recently passed
Got what? Manikandan got what?
Net exam. Clear net exam. Passed by net exam.
Sorry, sir. I am breaking here. I'm not hearing properly.
He passed the ICEH net exam.
Which one? Oh, he passed. Oh, nice exam. Good. I can feel he has good knowledge. He has good sense to learn a lot of things. Yeah, keep it up, Manikandan. Good job.
The people said taking weekly.
I'm sorry.
Okay, with this, can you mind of this now? I mean, there is no question. You are elaborate explanation of the regulation, the nuclear system, everything, but very nice and this able.
Sir.
Yes. No, I'm breaking the voice here. I'm not hearing the audio properly. Good. Everything is good, sir? Yeah. Any questions, sir?
No, there is no question.
Okay, sir. Thank you. Thanks for inviting. It's my honor to present here where I started my first molecular biology with you. I really liked the spark from Dr. Sheikh Umar Shahid sir, SS sir. So you are my first molecular biology teacher. Thank you. He took me to the MK University. So there only I wanted to do at least this kind of lab, but I went to bigger lab and I did a lot of work. I learned a lot in molecular biology. I would say I'm a good molecular biologist now. Thank you, sir. Thanks for the spark in me, finding a spark in me. Thank you.
Okay. Yes.
Okay, I'm good to go, sir.
Okay.
It's 1:40 here. Thank you. Thank you. Awesome. Thank you, sir. Bye, Puhari. Puhari is there. Is Puhari is there? Say hi to Puhari. Pugari is my classmate, batchmate. Hello, is it Pugari?
No, this is Suri.
Oh, Suri Narayan. Oh, hello, Anna. How are you?
How are you?
Thank you, Anna. Thanks for. I saw you are also giving talk. All the best. I'm sorry, I will go to sleep. I didn't the whole day. I had.
Yeah, completely understood. Thank you for your valuable time here. Thank you.
Yeah, thank you, Anna. Nice meeting you. Say hi to our friends.
Yeah,
I miss.
sure, sure. Thank
I
you.
miss Bharatiya University also. You know that.
Yes, yes.
Bye.
Bye.
Bye,
Have a
Anna.
good sleep. Bye. Yeah.
Oh, patient.
Slide. This is another manual. Next. Slide move.
Topic.
Very good evening. Another tea break curriculum from the other, but before that tea break, we are having another session. So after this session, we can end up this. Yeah, so don't worry. Will be served with a very high tea and snacks. So in this technical session of today's last technical session, we have Dr. S. Suri Narayan, sir, Deputy Director and Research, Vinayagar Missions Research Foundation, Salem. So he completed his masters and the Master of Philosophy in Zoology at Jamal College. So he is our illustrious alumni, and he completed his PhD in Environmental Sciences from Bharatiya University. Prior to this position, he was a postdoctoral fellow at the University of Turin, Italy, and then the project coordinator, faculty, and also head in charge of Department of Water and Air. JSS Academy of Higher Education and Research, Dean University, Mysore. He also served as a science officer for the Scientific Committee on Problems of Environment, Scope and Research Center for Eco Environmental Sciences, aging from April 2015 to March 2017. He also a nodal officer for Scope India activities under JSS ACR, and he is a visiting professor at Pontian University, Thailand. Dr. Suri Narayan is in environmental monitoring, radiation ecology, and environmental microbiology. He also received of young scientist grant award on the Science and Engineering Research Board, served DSP, Government of India. He also received a visiting scientist fellowship from Chinese Academy of Sciences in 2016-17. He has been a chairperson, participant, and presenter at many national and international conferences, as well as an editorial board member for peer-reviewed journals. Sir has more than 40 published research articles to his credit, having HTS of 18 in Scopus. He edited 4 books in the field of environmental sustainability, carbon sequestration, environmental monitoring, and he published 5 book chapters. He is presently as an associate editor for environmental development and Elsevier journal. So we are all feel very happy and delighted inviting Dr. Surya Narayanan sir for this afternoon session. So on behalf of the organizing committee, we invite him to give his ideas and views on the topic implications of microplastics. So please.
Very good evening to one and all present here. Thank you, Dr. Zoology, for kind introduction. Thank you, Professor Sexer. Thank you, sir. Also, and sir, the faculty members of this department and the staff members of this department and the participants and students of this department of Zoology. It is my great pleasure and honor to be here in this department. It is always my pleasure and happy to visit the department. This is my first time I'm delivering the talk at the invitation of the organizing committee. I thank the organizing committee members for giving this opportunity. Topic environmental and health impacts of microplastics. Here I would like to give overview of microplastics. So what is microplastics? What is the type of what are the different types of microplastics? What is the sources of these microplastics and how it is impacting the environment and health? So this is an overview based on the literature studies done elsewhere in the world. Next. So plastics everywhere, you know that, right? Actually, we can say that we are living in between the plastics. You can see the plastics within vicinity of our present position. Within 5 meters, you can see at least one single plastic object, right? So these plastics are producing year by year. The production of plastics are increasing over the years, and also the consumption of the plastics also increasing over the years. Next is. So if you see these statistics by UNEP, United Nations Environmental Program 2021 data, here you can see the world production of plastics in 2020 is about 400 million metric tons of plastics. This is expected in 2050 around 1,000 metric tons of plastics. So the exponential growth, you can see how much plastics we are going to produce in the near future. Also, from 1950 to 2017, the world has produced nearly 9,200 million tons of plastics, in which 50% of the plastics were discarded as waste. Nearly 1,000 million metric tons were incinerated or burned in the economy. And also, there are 700 million metric tons of plastics were recycled, and 2,900 million tons of plastics are still in use. So you can see the exponential growth of plastic industry's production. We are here in 2020, in 2050, we are reaching 1,000 million metric tons of plastics. So we have to accept that we are not going to avoid the plastic. We are going to live with the plastics, right? Next slide. We see the production of plastics in the region wise. Asia is contributing the mostly 50% of the plastic production, and followed by the America 23%, and Europe 18.5%, and Middle East and Africa nearly 8%, and Commonwealth of Independent States, the former Soviet Union, is about 3% of plastic production. Next slide. We see the waste generation from the plastics. In 2019, data shows that China alone produced the waste of plastics, 2.5 crore tons of plastic. Whereas the United States produced 1.7 crore tons of plastics, and India, we produced 56 lakhs of tons plastics as a waste. Next slide. So we are all aware that plastic is a polymer, very strong polymer, and based on the polymer characteristics, we are classifying into polyvinyl chloride, polyethylene, polypropylene, and so on. But based on the size of the plastic, these plastics can be classified into categories, such as microplastic, mesoplastic, plasticals, and microplastic, mini microplastics, and nanoplastic. Any plastic equal to 25 millimeter or more than 25 millimeter is called megaplastics. So that is large, all the large plastic particles, plastic objects, and mesoplastic which is ranges from 5 millimeter to 5 millimeter. You see the microplastic, any plastic less than the 5 millimeter to 1 millimeter in size is called microplastic. Then 1 millimeter to 1 micrometer size of plastic is called mini microplastics. Less than 1 micrometer is called nanoplastic. This is the classification based on the size of the plastic. Next slide. So all these days we have a debate on the size of the microplastic. Whereas in 2017, NOAA, National Ocean and Atmospheric Administration, they defined the microplastic. Any plastic that is less than 5 millimeters is called microplastic. Next one. So microplastics can be plastic pellets, microbeads in the personal care products, and microplastics in the paints, textile fabric industries, sewage treatment effluents, sports grounds, vehicle tire gears, municipal debris, fishing gears, farming fluids in agricultural fields, construction industries, fishing nets, etc. Microplastics. Next. So these microplastics basically classified into two categories: primary microplastics and secondary microplastics. Primary microplastics are directly commercially produced microplastics, the small particles, which is released in the environment. The secondary secondary microplastics, which is originates from the large pieces of plastics, because the large pieces of plastic materials are still divided into smaller particles, is called secondary microplastics. Next slide. You can see the sources of microplastics, these microplastics. Next slide. Next slide. These are the sources, major sources of the primary microplastics. Tires used by rubbers as well as the plastic materials, synthetic textiles, what the clothes they are wearing, the marine coatings, because of the anti-corrosion activities, plastic coatings are used in the ships, and road markings, personal care products, what we are using day-to-day basis, the shampoos, shower gels, scrubbers, all these are having the microplastics. Next, plastic pellets and the city dust are the primary sources of microplastics. Next slide. The formation of the secondary microplastics, what I have said before, the large particles, large size of plastics are disseminated to smaller particles called secondary microplastics. This can be derived from the three different processes: physical and chemical and biological process. Physical process is nothing but the mechanical process through the wind or naturally it is disseminated to smaller particles. It's called a primary physical process for microplastics production. And chemical process can be the weakening the polymer bands through the heat, thermodegradation, thermal oxidation, water hydrolysis process, and ultra degradation, photodegradation are fragmented into major plastic to microplastic to microplastic. And biological degradation using the microorganisms. So microorganisms can digest the larger size microplastics into the smaller size microplastics. So these are the 5 different types of microplastics commonly used: fibers, pellets, fragments, microbeads, and the foam. Fibers normally come from the clothing. Fragments that I said, the fragments of large plastics. Foams which are used in the coffee cups and food packaging materials as well as the thermocols. And pellets directly manufactured from the industries, and microbeads which are used in the personal care products. That is what I said, the scrubbers, facial creams, or the shampoos and soap. So these microplastics, through fragmentation, the degradation and incineration in the environment. Next slide. This is sources of microplastics. This picture shows the entire sources of the microplastics. How the microplastics originates and where it ends. There are two different sources: direct sources and diffuse sources. Direct sources through municipalities, industries, and construction industries, through the personal care products industries, and the plastic manufacturing bags, which is consumed by the consumers, that is the public, and the used products will be discarded as a waste. It will reach to the water environment through the municipal treatment plants or municipal waste, or directly from the industries as the leftover products, and through the landfills also. All these microplastics are reaching to the water bodies, and through the water bodies it is ended in the marine ecosystem. In the marine ecosystem also, through the coastal activities, tourism, and shipping and cargo fisheries, these are all contributing the plastic pollution in the marine environment. Next slide. So these are the potential accumulation sources of microplastics. You can see the microplastics, the surface layers, and the water column. Based on the density, you can see the microplastics in the water column, and seabeds based on the densities deposited in the seabeds, and you can see the plastic materials around the beaches wherever you. Next slide. So statistics says that there are 51 billion of microplastics are floating in our seas. For an example, in the Mediterranean Sea, which is surrounded by the European countries and the northern part of Africa, that the sea is alone receiving 17,600 tons of waste per year. So you can see the amount of plastics which is deposited in the marine environment. Next slide. This is an interesting map. You can see the global distribution of microplastics. How these microplastics is accumulating through the ocean currents. It is through depicts the ocean currents. Here you can see the Asia region, and in front you can see North America and South America regions. So these ocean currents, this picture depicts the color difference from white to thick red. Thick red denotes the highest concentration, which is about 10 kilogram per square kilometer. So you can see here the big pads like an ocean. The microplastics, all the plastic materials are floating here in Pacific Oceans, look like an island. This is called the Great Pacific Garbage Patch. So this is floating nearby the Pacific Ocean, between the Miami and the California. There are about the size of this garbage is about 1.6 million square kilometers. It is the size of 3 times higher than the France country, and half of the Indian continent, Indian landscape. So the scientists found that there are 1.8 million pieces of plastic floating in this garbage, and here most of the 94% of the objects are belongs to the microplastics. These waves are coming from the Japan, Mexico, Taiwan, China, Philippines, Canada, Chile, Colombia, Germany, Italy, Korea, and Venezuela. All these regions surrounded by the Asia Pacific region, as well as the America and the upper part of this. They are coming. All these plastics are through the ocean current. They are depositing and they are accumulating in the Pacific Ocean. About 700 species have been affected because of this garbage, and 100,000 species are propagating because of these plastic materials. 84% of these plastics contains the toxic substances which is harm to the marine animals. So here, the oldest object in this garbage was found varying from 1977. The concentration of plastic increases exponentially every year. So here you can see the portion of this garbage floating island of plastic in the Pacific Ocean. Some of the scientists are still working to characterize this in the waste. Next slide. So based on that, we have done a study in collaboration with the Concord University. A study on occurrence and impacts of sources of microplastic in the marine environment of South China Sea, Gulf of Thailand. South China Sea and Gulf of Thailand is the hotspot of receiving the plastic materials in huge quantities because of these two rivers, Pearl River in South China and Mekong River. The Pearl River originates from the South China and it ends near Taiwan. And Mekong River originates from the Laos. It flows to the Thailand and to Vietnam and Cambodia, it flows into the Gulf of Thailand. These two regions are economically as well as ecologically very important hotspots because of extensive ship activities and the commercial activities. This is the most important place we have to analyze. So that in view of this, we have gone through a rigorous of review of studies. There are 34 sites frequently detected in the microplastics. We found that these microplastics in water, sediment, fish and chlorine, mangroves, etc. and all the environmental components, they identify the microplastics in this region. Most of the microplastics belong to the fibers and fragments and the polyethylene. So this paper recently got accepted by the research journal of physics and environment. We published in 2020. Next. So where, what is the sources? How these plastics are coming into the marine environment? The distribution sources of microplastics in the world is by the IUCN was found that 35% of these microplastics comes from the synthetic textiles, and 28% from the car tires, and 24% from the city dust, and road markings, direct coatings, and the personal care products 2%, and plastic products about 0.3%. All these are major sources for pollution in the marine. Next slide. So when we look at into the polymer characteristics of these plastic materials, they found that 54% of this marine debris comes to the polyethylene, and 16% belongs to the polypropylene, and the 30% plastic found in polystyrene. Next slide. So what is the status of current microplastic research? This is an interesting slide you can see here. The publications of microplastics started from 2012, and slowly the research number of research are increasing over the years. You see, 2020 and 19, nearly 1,000 articles are coming out from the different parts of the world. So you see the number of publications, the two deficiency marine publications, marine related research. So the number of researchers are higher than the terrestrial environment. So there is an ample opportunity for all the researchers and the environmentalists to work on the terrestrial environment on microplastics. Here, this is the map of how the people are networking, scientists networking each other on microplastics. Next slide. So coming to the methods and the characteristics of microplastics, scientists are normally using the methods of separate methods using three salt solutions: sodium chloride, sodium iodide, and zinc chloride methods. Among these three methods, the zinc chloride is the prominent method, which is recovering the 93% of more than 93% of the microplastics. These microplastics also been detected in 2019. There is one method developed by Graphing et al. 2019, magnetic extraction of hydrophilic iron nanoparticles that find the plastics. This shows the 93% of the recovery of microplastics. So as of now, there is no standard method for extraction of microplastics. So because of the technical difficulties to extract 100% microplastics from the different environments. So that is why there is no standardized method for extraction of microplastics. There is an ample opportunity for the researchers to standardize method and to come with the very accurate method for segregation of microplastics in different samples. Next slide. So, with regard to the measurement, identification of plastic is measured visually by the size or chemically using wide variety of tools such as filtration, visual review, microscopy, mass spectrometry, infrared or Raman spectrometry. These are the methods used for characterizing the microplastics. And these can be measured in the units particle counts versus gram or liters. Next slide. So this is a very interesting picture you can see here. As I said, micro or nanoplastics are similar size of many microorganisms. When the size is getting smaller, then the analysis expense is also lower. So you see 1 micrometer to 10 nanometer particles can be accessed to the gas chromatography, mass spectrometry. This equipment alone costs about 200,000 to 300,000 US. So see the expenses of this analysis. And from 1 micrometer to 10 micrometer, the particles can be accessed either through ATR or micro-Raman spectrometer, which costs about more than 50,000 US. That is why when the size is getting smaller, like micro-Raman, the analysis cost is also getting high. Next slide. Now we can see the impacts in the environment of the microplastic impacts on the next slide. So as I said, microplastics have been detected in all the environmental compartments, even in the far-reaching places. Some of the microplastics are detected in the Arctic region. Some of the microplastics detected in alpine mountains in the high altitude. and even in the deep sea sediments also, microplastics can be detected. Uneven distribution of microplastics because of the human activity with the different environmental factors such as waves, current, tides, wind directions, the river hydrodynamics, water density. The concentration and character of microplastic can be highly variable, spatially temporal. That is why we cannot give any assumption or any conclusion based on the reports published elsewhere. So there is an ample opportunity. We have to analyze, we have to measure the microplastics availability in the environment region-wide. So that is why there are a lot of opportunities still exist to work on the microplastics. Next slide. When come to the agricultural ecosystem on soil, the microplastics normally come to the agricultural nearby dumping ground. There are garbages. These microplastics are degraded or disintegrated smaller particles that is reaching the soil. As well as mulching of plastics, you can see in the agricultural land in the temporal country, they are using the mulching plastic thin layered soil, which prevents the soil evaporation, which prevents the soil erosion. So these temperate countries are using this mulch agricultural farming methods. So these are the small thin layer of plastic for the plastic covers. Normally, what the farmers will do over the period of time, these microfilms will be photo degraded and dissected into smaller pieces. They are not removing these microplastic, these plastic materials. Next harvesting, they are clubbing with the same plastic materials. So in that way, it is reaching to the soil environment. Similarly, from the wastewater irrigation, the microplastics are reaching to the agricultural land because of no treatment plants having the proper facilities to remove the microplastics. And these microplastics also can be deposited through the area deposits. So in this way, the microplastics are reaching to the agricultural land as well as the soil profile. So we see when it reaches to the soil profile, it directly affecting the soil pH and also affecting the soil microorganisms. When the soil pH and soil microorganisms are affected, these properties can also be affected. Soil nutrient balance, soil porosity, soil fertility, soil aggregate stability, soil bulk density, soil enzyme activity, water holding quality. With regard to the microbiome, increased mortality rate can be done in shown in soil fauna, acidity stress, moderate soil microbiome, decrease the soil biological and vigilance activity. Why I am saying here, all these activities still yet to be proved because no such studies on these effects. So there is an ample opportunity for the researchers who are working in microplastics. They can explore these possibilities and they can work on this soil plant interaction system in the agricultural ecosystem. Similarly, how this is affecting the yields, how it is affecting the plant growth, how it is increasing phytotoxicity, how it is decreasing the nutrient content, all these things are still unexplored. Many people want to do research in microplastics, they can explore these opportunities. Next slide. Coming to the air, we are every day we are inhaling the microplastics, even in the indoor environment as well as the outdoor environment. Clothing materials, washing, drying, deterioration of house furniture, we are inhaling as a microbeads, and from the external environment, we are inhaling as a microplastic fibers. We are inhaling and it will create the cytotoxicity and inflammation. Next slide. Even water also, it is contaminating like anything because these microplastics are the carriers of the contaminants. You can see the cocktail of contaminants. Microplastics is the cocktail of contaminants because of this physical and chemical nature, like the hydrophobic and the hydrophilic interaction, and the hydrogen bonding, water forces, pore filling capacities. All these qualities are attracting the contaminants towards the microplastics. So that these microplastics can acting as a vector or a carrier to pass the contaminants from one place to another place and polluting the water environment. Next slide. Similarly, heavy metals also attracted towards the microplastics either by resorption or absorption based on the chemical characteristics. This also microplastics carry the heavy metal contents from one place to another place and contaminating the water environment. Next slide. You can see the new word for the here for the last 2 years. What is called the plastic sphere is nothing but the compression of microbial on plastic debris. Like the ionosphere, thermosphere, lithosphere, photosphere. This is also the plastic sphere you are going to hear in the future. So this plastics materials also carrying the pathogenic organisms, microbial organisms, saprotrophs, heterotrophs, phototrophs, predators, and symbiotes. Next slide. What are the health effects of the microplastics? Next slide. Do you know how much plastic we are consuming? A study conducted by the University of Newcastle, Australia, they found that every week we are consuming 5 grams of plastic. Every month we are consuming 21 grams of plastic. In our entire life, we are consuming nearly 20 kilograms of microplastics. So you can imagine that knowingly, unknowingly, we are ingesting the plastics. Next slide. How we eat, how we drink, how we breathe into microplastic. A study conducted in the based on the American diet, they found that in the bottled water of 1 liter having 94 particles of microplastic. In the beer, 32. There we breathe 9 particles. And in the air water, 4 particles, seafood 1 particle, and in the sugar 0.44 gram, 44 particles gram. So see how much plastic we are ingesting through different food sources. Next slide. So this is the effects like other contaminants. Microplastics can also form the behavioral effect, morphological effects, and the reproductive effects. Next slide. Recently, the biomolecular studies are coming out. Because I said 2012 onwards only, these microplastic research came into the limelight. Now, the biomolecular studies also being conducted. The recent studies by 2022 by Zoology from Mexico, they found 20 biomolecular studies abundance of microplastics in 16 different types of human biological samples. They found 700 nanometers to 5 millimeters smaller particles in the majority of them belong to 5% fragment microplastic. Next slide. Next slide. So this is the review of literature published recently last month in 2023. The different studies conducted by different studies conducted throughout the world. They consolidated the report and they published in 2023. So that overall annual intake of microplastics, a person might exist in 39,000 to 52,000 microplastic particles per year. It is expected to increase in the coming years, 74,000 to 1,21,000 microplastic particles per year. So imagine that how much plastic we are going to ingest. Similarly, the blood samples also found with the microplastic 1 microgram per milliliter, and breast milk also found in the microplastic 0.72 microplastic per gram. Even in the apical materials also 1 to 138 microplastics per gram was identified. If you see the lungs also 1.17 to 2.84 microplastics per gram were identified. You see the saliva, even in our saliva also 0.33 microplastics per individual was detected. Next slide. Microplastics in the food. The same review study conducted in different parts of the world. In review of 77 studies of microplastics in various food samples, which is approximately 140,000 to 155,000 microplastics per year, which is comes from the different parts of different food materials. You see the seafood in pieces, it ranges from 0.1 to 8.6 per gram in fish, and in the shellfish 10 to 100 microplastics per gram in oysters and mussels. These are the plastics found in the fish samples as well as the oysters and other crustacean samples. You see the salt content, how much plastic is there. In the sea salt, it is ranging from 0 to 1,600 microplastics per kilogram. In the dead salt, it is ranging from 8 to 462 microplastics per kilogram. And in the rock salt, ranges from 0 to 204 microplastics per kilogram. We see we are consuming the plastic even through the salt what we are using the day-to-day life. The drinks and the beverages, microplastics have been detected in the beverages such as apple juice, wine, milk, and beer, ranging from 2 to 254 microplastic particles per year. Next slide. So these microplastics can also found in the animal and the plant species, like from plant anchovies, oyster, mussels, tortoise, report, mice, and chickens. They found these microplastics. In studies among 800 animal species, 244 species were contaminated with plastic, and 220 species were found in the plastic particles. Even in the plant species, you can see the freshwater algae, they have coral, duckweed, agricultural plants like tomato, cucumber, carrots, lettuce are linked with the microplastic. Next time. What is the status in the aquatic environment? You see in the marine water, the sample surface, the sea surface will contain an average range of 0.02 to 8,600 microplastic items in a cubic meter. If you see in the sediment samples, it is ranges from 185 to 80,000 microplastic items per cubic meter. See how much plastics are accumulated, deposited in the sediments of the sea. Freshwater samples, they found that 1 particle per 100 cubic meters and 1 million particles per cubic meter and 100,000 particles per cubic meter along the 4 lines of North America, Europe, Asia, and Australia. So everywhere, plastic microplastic has been detected. Even in the drinking water, European samples shows that the tap water range from 0 to 628 particles of microplastics, and in the bottled water, 0 to 4,800 particles of per liter microplastics found. The wastewater in the US, United States, the wastewater treatment also releasing 50,000 to 50 million particles per day in the environment. Next slide. Similarly, microplastics found in the soil as well as the air, indoor and outdoor. Next slide. When coming to the biological system, there are many studies that have been conducted in the animal experiment, like what is the effect on the digestive system. We see Ben et al. 2022, he subjected 49 mice samples with the 5 micrometer of polystyrene. He ingested these polystyrene for 90 days in the concentration of 100 microgram per liter and 1000 microgram per liter. The result shows that drop or microanatomic colon effects and also changes in the cell proliferation, cell death or nutrient supply, chronic inflammation and oxidative distress. With regard to the digestive system, next slide. You see in the reproductive system, there are the studies that conducted in the animal model. You see the Jamal, he conducted 105 male mice species with the injection of 0.5 micrometer polished surface to 180 days. The results found that the male reproductive failures has been detected, like alteration in the production of reproductive hormones also detected. Next slide. There are studies on the respiratory system. Again, it is in the animal samples. So far, no studies in the human samples. That is why I'm showing all these slides. There is an ample opportunity to study the biological system effects on microplastics. You see the respiratory system effects, C et al. 2022, he subjected 36 mice species 3 times in a week for 3 weeks from 1.25 microgram to 6.25 microgram concentration. They found that pulmonary fibrosis, alveolar epithelial and detection of oxidative distress. So the evidence shows that respiratory inflammation also occurs through the microplastic. Next slide. So these are some of the pictures for your interested. See how the microplastics is sedimented, how microplastics are floating in the surface water. It reaches to the marine environment, and many sea water animals species like mollusks and bivalves, and the fish species predators are often with the microplastics. You see this picture, the water media with the microplastic particles in its guts in the white dots here, and even for the fin whales and sharks also infected with the microplastic. Next slide. This is the bird died because of the ingestion of microplastics. They found that flame retardants, benzoyl UV stabilizers found in the guts and the lungs of the birds. Similarly, whales and the beneficial organisms of the fish is also affected by the microplastic. Next slide. This is a picture by Cole et al. 2013. You can see the zooplanktons ingested the microplastics in the green color. Similarly, this bird died because of the microplastic ingestion. The scientists extracted this microplastic from the guts and the lungs. Next slide. So, as I said before, there are not much studies on the human system. Recently, in 2022 December, one study was conducted by the group of people. They found that out of 6 placentas, they found 12 particles of microplastic, 4 of the placentas. So, human tissues also accumulating the microplastics. Next slide. So, it is clearly evident that there is a possible effects on neurological, cardiovascular, respiratory, renal, and gastrointestinal based on the literature review what we have found. So we have to be very cautious and we have to give more attention on towards these microplastic pollution and the combating the microplastic pollution. Next slide. So mitigation, so what are the mitigation methods? Many countries are coming forward to implement many regulations to combat this plastic pollution. Next slide. So one of the key development recently held in 2022 along with India, 174 nations are agreed to commit to develop a legally binding global agreement. With this agreement, these countries can address the alternatives to address the full cycle of plastics to design and reusable and recyclable product material. And also they need for enhanced international collaboration to facilitate access to the technology, capacity building training program, scientific and technical cooperation is expected through this agreement. This agreement will be finalized in 2024. Many countries are willing to sign this agreement to combat the microplastic pollution. So here you can see there is a lot of opportunities for collaboration nationally, internationally, and there will be a possibility of more grants on microplastic research. Next slide. So these are the international treaties proposed by the United Nations. Many countries are voluntarily signed the agreement to combat the plastic pollution. Next slide. So one of the key development in 2019 conference of the parties by Basel Convention by United Nations calls upon the parties and others to promote environmentally sound and efficient management of plastic waste to reduce the discharge of microplastic. After that, 2022, there is a draft agreement is coming into the effect. They are going to sign in 2022. Next slide. So many countries, as I said, voluntarily they are coming forward to combat the plastic, like Norway and European Union. They are planning to ban the microbeads and microfiber productions, and also they want to phase out manner. They want to stop the productions in the near future. Next slide. So what is the status in India on the mitigation methods? We have the Environmental Protection Act 1986. In this, section 3 and 6 and 8 and 25 helps us to prevent this plastic pollution. Similarly, plastic management and handling rules was implemented in 2011, which is very specified regional conditions of carry bags and plastic waste management rules has been framed. And in 2021, draft of plastic waste management was proposed by the government of India. It proposes to prohibit the manufacture, import, stocking, distribution, sale, and use of earbuds, plastic sticks for balloons, plastic flags, candy sticks, ice cream sticks, polystyrene, thermocol, decoration, and any other single-use plastic commodities from 1st January 2022. It also proposes to prohibit the manufacture, import, stocking, distribution, and use of single plastic commodities. That is why you have seen many places this is banned. The single-use plastic has been banned, and this circular has been sent to all the stakeholders and the administrators to take necessary steps to prevent the single-use plastic. So this is implemented in India. So many countries are voluntarily involving to ban the single-use plastic. That's why today morning also the news came in Vilupuram. They found the single plastic and they find the shop owner 25,000 rupees fine. Today morning news came. Next slide. So this is the map showing that how many countries are banning the single-use plastic. It depicts in the green color. This shows the positive sign. Somehow we are initiated to prevent the plastic pollution. Next slide. So conclusion, the environment and the health impacts of microplastics is growing concern that requires immediate attention. The action to reduce and prevent further harm to the environment and human health. I said there are a lot of things that we explore. The scattered data is the major concern, and we have to collect all the data available for better management of these plastics. And there is a need for research in the terrestrial environment, freshwater and the air, because most of the studies focusing on the marine environment, and there is no such studies on cellular uptake, how the inflammation occurs, oxidative stress, hormonal changes, and genomic instability. So all these things are open for researchers and scientists. They can explore the possibilities on microplastics and the effects on the cellular mechanism. So also policy to be made on the region based, because developing countries and developed countries they need separate laws and regulations. Accordingly, the policy can be revised with the researchers and it can be implemented in the future. Next. These are my references, what I refer. I would like to inform these participants that we are organizing the international workshop on combating plastic pollution in terrestrial environment on coming 14th and 15th in Mysuru, in collaboration with the NAM SNT countries, Delhi, as well as Scope Environmental Organization from Netherlands and with the JSS University Mysore. This registration is open. The delegates from 15 countries are coming physically. They are sharing their knowledge. They are giving their experiences on plastic pollution from 14th and 15th of March this month. The registration is open. I'll give this contact information to the organizers and the secretary. If you want to participate, you are interested, you want to listen or you want to experience with the lab countries how they are handling the plastics, you are most welcome to participate in this workshop. Next slide. So with this, I would like to conclude my session with the saying of Sri Amit Ray. He is the idea of compensate artificial intelligence. He said that plastic pollution free world is not a choice, but a commitment to life, a commitment to the next generation. It is our responsibility as academicians, as the students, as the researchers, and the scientists. We have to commit with ourselves to save the nature for the future generation for the sustainable world. Thank you. Thank you all.
Thank you, sir. Now, students can make use of this opportunity of your clarification.
So, like you, every week or every month, you have a research article says that you have found microplastics in blood, chest milk, etc. But each article they say that we don't know how it affects the human body. They are telling what is the common factor regarding why you can't find the effect on human.
That is why I said in the presentation, this is the cost gap in the research. The accumulation of identified, the percent of microplastics are identified, but how it is interacting with the cell, whether it is causing the cellular there is a lot of opportunities for the scientists to identify the cellular mechanism and hydroactivity of the Tactics, practically saying, if my knowledge goes the last 20 years, we are talking about the plastic pollution. But are we stopping the use of plastic? So that is why there is an awareness required. I hope the scientists will find but avoiding the plastics, as well my knowledge goes, I don't know how far it is possible. So being an educational citizen, educated citizen, we have to make ourselves try to avoid as much as possible use of that.
We are using plastic for the highly for the past 30 years for the before that what the data available for microplastic. That time also we are using plastic.
Only this microplastic research started doing because of the research articles we found that 2005 there is one article. After that 2010 only they started exploring the microplastics. So before that there is no that data on microplastics.
We are using the medical plastic even in class 1, and everything. So something like nylon, so that was very small.
There is no doubt. We are on the plastic in our body, but we don't know yet how far it is affecting our cells of human cells or animals. So that is the big gap we have to address. Anybody interested to do microplastic research? Anyone interested to do microplastic research? Thank you, sir.
So really very useful session. Behalf of organizing team, I convey my sincere thanks to our chief guest today, Dr. Surya Narayanan sir, for enlightening us regarding the microplastics and research in this particular microplastic. So now I welcome our organizing secretary to Adana Shaw to our today's chief guest.
Thank you, sir. Ladies and gentlemen, hope you all make use of our today's various technical sessions. Now, we will wind up our today's session. Thank you all. The refreshments will be provided outside of the hall.
Be there tomorrow.